present here, as well as our PhD scholars. Welcome to the vibrant campus of MAHE Bengaluru, as we celebrate National Science Day again in 2025. We celebrate National Science Day remembering Professor V Raman. He was one of the most influential Indian scientists, and his discovery of the Raman effect won the Nobel Prize in Physics in 1930. But his contributions are not singular. He has a lot of different contributions to Indian science. In addition to the Raman effect, he also was key in developing Indian scientific institutions. He helped set up the Indian Institute of Science, IISc, from where our keynote speaker comes from, as well as later he founded the Raman Research Institute in 1948. He was also very important in setting up optics, acoustics, and crystal dynamics applications in physics and engineering in India. And in fact, he was the first Indian director of the IISc post the British era. His contributions are innumerable if we think about fostering the young scientists in India. His mentorship include a lot of different scientists, but one of the most famous mentees included N Rangarajan of the Ramachandran plot, which we all know from structural biology. He believed in cultivating curiosity, independence in research, encouraging students to actually challenge the existing scientific paradigm. I've already mentioned about his contribution in setting up scientific institutions, but he actively promoted science education in India, frequently giving public lectures and encouraging schools and universities to focus on science. His impact is still felt today in India in our approaches to scientific research and education. This is a golden opportunity to enrich our minds and spirits, learning from all the esteemed scientists present today. Now I request our dignitaries, Professor Madu and Professor Rangarajan, as well as Professor Jyoti, to please come on the stage and open our program by lighting the lamp. Thank you to our dignitaries. I now invite our Dean, Professor Jyoti, to officially introduce National Science Day and our guest of honor, Professor Madu, and also introduce our guest of honor, Professor Madu V Raman. Professor Jyoti.
A very good morning to everybody. I would have expected a more crowded auditorium even though, right? So I think it's not yet good morning for many. So with this, I would, nonetheless, I think people will keep joining. I take this opportunity to welcome our distinguished guest for today, Professor Madu V Raman, the Pro-VC of MAHE Bangalore. He had been a champion of research and especially of MIRM over all these years. I'd also extend a warm welcome to our very beloved PNR, which we used to call him then, if I can take the liberty. Professor PNR is an eminent scientist who had done a lot of research on vaccines and eukaryotic gene expression. Welcome you, sir. So every year on Science Day, we have a theme, right? This year, the theme of the Science Day is Empowering Youth for Global Leadership in Science and Technology. It's a very, very big line. But what is leadership? I was just thinking, what is leadership in any realm? It could be science, it could be industry, or it could be anything. So what is leader? A leader is not just a position. Most people confuse leadership to a position or a title, but in fact it's not. It's a role. A leadership is a role to motivate a huge mass of people so that they can take the baton forward, right? So what should be the qualities of a leader? We are talking about empowering youth to become leaders. So what should be the leader? What should be the quality of a leader so that they can empower many more people, right? So a leader should himself or herself should be motivated enough. Then only they can motivate a team, right? The second facet which I was thinking about leadership is integrity. Integrity in whatever the person does, because then it becomes a norm that you have to function this way with all the people who are following him or looking up to him or her. What is the third quality of the leader? A third quality of the leader, which I feel, is you have to mentor a student so that they become better than you. So the leader should be least insecure, right? So leadership is a huge, huge responsibility. And history says that great leaders are the ones who have built better leaders than them. A good example is the V Raman K.S. Krishnan story, where K.S. Krishnan was a student, and the contribution of V Raman as well as K.S. Krishnan is equally important in the field of physics. So when we talk about science as a profession, I don't think science is a profession, by the way. What is science? What is research as such? So researchers can just not be slotted into a professional person. It's because research thrives on inquiry. It thrives on curiosity to know something, right? And it thrives on the perseverance to follow that curiosity with logic, with experimentation, test it, and that's where you satiate your curiosity. Science has to be done and is done for the sake of science and not for the sake of titles. A simple example is that of V Raman, where he was just observing light falling on a blue sea, and a simple curiosity as to why the light is scattering tickled his mind, and he pursued it in the lab. He kept on pondering about it, and his dedicated pursuit to his inquiry had led to what we know as the V Raman effect, or what eventually led to the Nobel Prize. So a leader need not generally be a scientist. It is not leadership or science in general is not restricted to a particular stream. A scientific way of thinking could be there in any field. It could be in accounting, it could be in philosophy, it could be in humanities, it could be in biology or any field. So with this, it's my privilege to welcome our chief guest, Professor Madu Veeraraghavan. He, apart from being an astute administrator, which most of us know, he's also a very, very passionate researcher. Professor Madu Veeraraghavan is a doctorate in finance with around 20 years of global experience in accounting and finance, and most of his publications are very well cited in his community. Apart from being an administrator, he is actually a researcher by heart. You can see very few pro-VCs of any organization listening to researchers, thesis guides, and research scholars and try to understand, like, what are their problems, how we can better them. And Professor Madhu does this from time to time, taking time from his busy schedule. He has been particularly supportive of MIRM being a research-intensive institute. Welcome you, sir. Right. Every year, we have a theme. Our VC tells us a theme. Last year, it was excellence in research. This time, it is fostering industry-academia collaboration. So when we were having the Science Day, we were making the list of whom we should call for the Science Day. We wanted somebody who had a very good academic background, has done very well in research, fundamental research, and also extrapolated it to a greater good. And the person who ranked, the first person whose name came in my mind was Professor Rangarajan, whom we call as PNR. So what started as understanding eukaryotic gene regulation in Pichia pastoris in his lab led to the hepatitis B vaccine, which he's mostly known for. So his career journey is a right amalgamation of academics, research, and innovation. Welcome you, sir. Right? So I think MAHE has been a trailblazer in many things, despite being a private university, and our VC, Professor Venkatesh, is a champion of sustainability. And when we talk about sustainability, you have to take care of the environment, the usage, trees. So this time, instead of having poster sessions, we have flash talks of students, PhD students. And sustainability is not just about paper and environment, it's also about time. Time is one dimension of sustainability, so I would not take too much of time in welcoming the dignitaries here. We can probably start off with the program. Welcome all of you.
I would now request our Pro VC, Professor Madu, to come and please enlighten us with some few words regarding the National Science Day. The podium is yours. Please take it.
Jyoti's already given me a warning that time is also sustainability stuff, so okay. Professor P. N. Rangarajan, they call you PVR, so greetings from MBR to PNR, from the Indian Institute of Science. He was the keynote speaker today. MAHE family members, colleagues, students, and friends. I would have loved to see a lot more people here today, but unfortunately, I don't know for what reason the students are not here. I hope that next Science Day we have a lot more people. So we're all gathered here to observe, maybe celebrate the 38th National Science Day, which means we're all together pay our respects to Professor V Raman, not only on his birth or death anniversary, but on the anniversary of the day on which he discovered a phenomenon that Jyoti also referred to, a phenomenon of light that's now named after him, the Raman effect. We all know a lot about Sir Raman, the scientist. Dr. Rangarajan will, of course, tell us more about Sir Raman. So I will focus more on Sir Raman, the man, not the scientist. The day the Raman effect was discovered was 28 February 1928. Just 2 years later, in 1930, he was awarded the Physics Nobel. That made him the first Indian to win any science Nobel. A quarter century later, he was awarded the Bharat Ratna. Please keep noting the timeline. I will come back to the timeline again, but a little bit later. So C.V. Raman passes away in 1970, about 15 years after his decorated with India's highest civilian award. 17 years later, some bulb in New Delhi lights up, and the maiden National Science Day is organized. In 1987, 1 year shy of 6 decades after the Raman effect is discovered. How I wish the Raman effect was a term in political science, not in physics. Can you believe it, that only political and social leaders were bestowed the Bharat Ratna in the first 5 years of the award's history? That's so far as much how this great nation honored Sir Raman on his scientific achievements. I often wonder, what would Sir Raman have thought of this had he been alive? To find an answer to the question, I take you back to the Bharat Ratna episode. Do you know that Sir Raman was not conferred the Bharat Ratna at any grand ceremony like it happens today at the Rashtrapati Bhavan? Instead, a postman delivered a medal and citation to his Bengaluru address. This happened because Sir Raman could not attend the grand ceremony as he was busy in Bengaluru assisting one of his doctoral students with his thesis. Imagine the grand old man of Indian science opting to be by his student's side than basking in the light of his glory. That was his commitment to science, to its growth, to its promotion. Therefore, my first lesson, or the first lesson, the science is important, the scientist is not. What then would have thought about the late Indian awards had he been today? He would have excused himself from every award function, attended instead the inauguration of some digital science library somewhere in rural India, or have been busy in determining how better to take science to the youngsters, or to farmers, or to far-flung remote tribes, other equally deserving people to benefit from it. To him, his service to science was its own reward. He left all his property to science education, something today's politicians would never do, or cannot do ever, rather. He simply did not care what he got from whom. He was deeply focused on what he could contribute, and that leads to the second lesson: science lovers can only be givers. True scientists never take. I was in school as a high school student when I was given an article written by Sir Raman for Presi writing. That article struck me for so many years that it inspired me to work hard. I remember Sir Raman raising a very pertinent question in that article. There would have been millions of people who would have seen things fall to the ground. Thousands of people would have been seen apples fall to the ground. So why was Isaac Newton the first to discover the gravity? Sir Raman's answer to that question stunned me. He wrote in the article that Newton was already immersed in thoughts, just what Jyoti was referring about Sir Raman, questions and answers about gravity. His immersion in what he was doing was merely triggered by the apple's fall. It was no fluke that Newton discovered gravity. It was an immersive effect of many, many years that led him to discover one of nature's fundamental forces. We can say the same for discovery Raman effect, can't we? Since that day, I give my all to any project I take up, and I must tell you that the habit may have burdened my health, but never my conscience, every single time. The final lesson: there is no such person as a lazy scientist. I urge science enthusiasts, students, scientists themselves, not to have wall clocks in their workspace. Science has never worked on flukes, it never will. Please make your immersive effort count. How many of us are aware of Article 51AH of our Constitution? Sarasu is not here from the law school, neither are any law students, so I'm sure none of you would know this. Article 51AH of the 42nd Amendment of 1976 clearly states that it's a fundamental duty, among others, of every Indian citizen, not just scientists, to develop scientific temper, humanism in the spirit of inquiry and reform. India continues to be the only country in the world to have the encouragement in its constitution. May we take this more seriously? I feel so. More so on the National Science Day. Before I spare you any further with my rant, I think I must put before you one more nugget from Sir Raman's life. Do you know that he was the topper of the Indian Finance Service examination of 1908 and was inducted into the service at the ripe age of 19 in Calcutta as an Assistant Auditor General? So you never know. One fine day, a professor of finance called Madu might actually end up discovering some as yet undiscovered phenomenon. I have high hopes of myself, and our Constitution also mandates it. The only thing is to start the immersive scientific effort. Thank you very much.
Thank you for those inspiring words, and we will definitely be keeping our eyes open for that really big discovery and maybe the next Nobel. And now I take this opportunity of the National Science Day to actually launch the newsletter of MIRM. I would again request Professor Madu to please come on the podium and launch virtually. We will again not be printing any newsletters this year, but we will be launching it on the screen. Yes, just, oh, careful. Just this arrow key and it will get.
I would like you to do that, please.
Yes, please. This arrow key, yeah, yep. Thank you so much. I would also like Professor Madhu to now felicitate all the contributors from the student side with a cash prize who wrote articles for this newsletter. I call upon Sridipta Rath, Ausapa Moreb, and Pratyush Sareen to come on stage to receive their certificates from Professor Madu. Please. Yeah, the other two students are and Professor Anujit. Maybe you can do the honors because I don't see the other students. Osama's there? Yeah, please, come. All of you, please come on the stage. Osama is here. As well as Pratyush, if he's here. Pratyush. To receive his certificate, he's not there. Yeah. Yeah. Yes. Okay. No, he's not here. Maybe Professor Arujit could just, because he is the also running the newsletter, so rightly he can take the certificate as well. Yes. Thank you so much to our dignitaries to launching our newsletter. We also take this occasion to felicitate our toppers. Yes, Professor Rangarajan. I would now request Professor Rangarajan and Professor Madu as well to facilitate the toppers of the Masters in Stem Cell Technology and Regenerative Biology. I call Ms. Raghavi, who came in first, and Ms. Nityashri, who came in second in the Stem Cell Technology and Regenerative Masters program. I would now like to call the toppers of the Masters in Translational Neuroscience program, Ms. Neetu, who came in first, and Ms. Trisha R., who came in second. Please come to the stage. Neetu is not here, so maybe. Trisha, I would request you to also take the certificate from Neetu. Yes. Thanks to our dignitaries. I would now like to request Professor Jyoti to please introduce our keynote speaker, and we would like to move ahead in the program to have his enlightening talk. Professor Jyoti.
Professor PNR, for me, he has always been the teacher who taught us gene regulation for a person who didn't know the D of DNA and R of RNA. So he will remain the same for me, despite being the dean of this organization. But for the world, PNR is known as an eminent biochemist and virologist who played a pivotal role in the development of the indigenous recombinant hepatitis B vaccine. Why his discovery was significant is, during the time when he had actually launched this vaccine, we were importing this vaccine from outside, and it was definitely not cost effective. He collaborated with Bharat Biotech back then in 1990s, where most of the researchers used to work in their own small labs. He thought about it one step further, and he thought that whatever you do in the lab should affect public health, or you should give it back to the society. So he collaborated with Bharat Biotech to create this vaccine, which was a significant achievement, as India was, as I told previously, importing this vaccine at a very high cost. So hepatitis B vaccines, which are being used in the universal immunization program from the government of India, are actually a discovery from his own lab. Let's give a huge round of applause for that. The recombinant hepatitis B vaccine developed by Dr. Rangarajan and his team has been a commercial success, and hundreds and millions of doses of these vaccines have been sold till date. Dr. Rangarajan's work in the field had a profound impact on public health in India, making the hepatitis B vaccine more accessible and affordable to the general public. His contributions to vaccine development and his research on eukaryotic gene expression has earned him numerous accolades. In fact, if I start reading out the number of national awards he has got, I think 5 minutes would not do justice. He had got the National Bioscience Award for Career Development in 2001 to 2003. He got the Swarna Jayanti Fellowship from DST. He got the Shanti Swarup Bhatnagar Award for Medical Sciences from CSIR in 2007. He got the Tata Innovation Fellowship from DBT in 2009 to 2014. And the JC, at present, he's a JC Bose Fellow from DST. So, but what is important also in his journey is he has sort of contributed in many ways as a researcher, as an educator, and as an innovator. So he has been, in fact, some of our students should look at the NPTEL courses which are there in the net, and he takes through the eukaryotic gene transcription right from what D of DNA and R of RNA is. And I still refer to his courses, sir. Right. With this, I would welcome Professor PNR to give his insights on his journey from the lab to the vaccine.
Check.
This can change the slides. Yes, it can. Just to move ahead. Okay. Will not be a pointer. Unfortunately. This is an LED.
Okay. Good morning, everyone. First of all, I'd like to thank Dr. Jyoti and Dr. Shweta for inviting me here to share some of our research work that's been going on. This year, July, I'll be completing 32 years of my journey as a faculty member at Indian Institute of Science. So whenever someone invites me to give a talk, it's also a time for me to go back and reminisce, what the hell have I done all these years? So I always take it as an opportunity to see, can I do some justice for my stay at this institute for this many years? So what I'm going to do in the next few minutes is to share with you my journey over the period of 3 decades, which is a mix of basic research, a indigenous product development, and also some innovative research that we have been doing. And these are important for me to share with you these days because today the expectations of the government from a researcher in a premier institutions are becoming very different than it was a few years ago. A researcher or a scientist in academic institution not only has to teach and motivate students, train them to be the future leaders, either in the academia or in the industry. He also is supposed to do world-class research, publish in high-quality peer-reviewed journals, contribute to knowledge generation in his area of research. At the same time, he also has a responsibility to contribute to the economic development of the country. It's now become, because we are not developed nation yet, we are still developing nation. We have to create jobs, we have to make products within the country so that it becomes affordable and it generates employment. So the government is actually looking forward to people in academia to also generate future leaders, future entrepreneurs, so that there are jobs created and products are available cheaper at much economical price. And we also have to innovate. Simply making things in India or manufacturing here is not enough. We also have to innovate. Take, for example, AI. Look what happened, what China did. The DeepSeek is an example, how it has disrupted the entire AI industry, and we are still not there. For many areas, India is still, we are followers and not the leaders. And this has to change if we have to become a developed nation in the years to come. Especially, we all talk about 2047 when India becomes 100 years as an independent country. So there are a lot of expectations, there are a lot of responsibilities, both from the teaching fraternity as well as from young people like you. So what I would like to share with you in the next few minutes is to see if I go back and then take you from the year 1998 onwards. I joined the institute in 1994, and what have I been doing in these areas of basic research, indigenous product development, and also an innovative process development that have come up recently, and have I done some justice as a researcher in a leading academic institution. Now, before I talk about these 4 vaccines, which is the title of my talk, let me just introduce you what a vaccine is. I am sure most of you have studied in the basic immunology course what a vaccine is. A vaccine is nothing but a non-disease causing mimic of a pathogen. So as you know, when a pathogen infects you, it causes disease. It can be a virus, it can be a bacterium, or it can be a parasite. But if you can somehow manipulate the pathogen, either by killing it or by modifying or introducing certain mutations in its genome, so that when it infects, it doesn't cause disease, but the antigens present in the pathogen is present to the immune system, so that the immune system develops both humoral as well as cell-mediated immune response, and therefore we get protection when the actual pathogen invades. So a vaccine is nothing but a non-disease causing mimic, and there are very many ways by which you can make the immune system fight the pathogen by giving a vaccine. A vaccine can be just an inactivated virus. This is the very traditional form of vaccination, where you simply take the pathogen, kill it using formalin, and then give it as a vaccine. The classical polio vaccine is an example, or the classical smallpox vaccine, which Edward Jenner discovered, is an example of the second one, where it is a live attenuated vaccine, where the pathogen is still live, but it is attenuated, and therefore it does not cause disease. And you also have what is called as the viral vector vaccines, where you take a antigen gene of that particular pathogen, put in another harmless virus and make it a viral vector, so that the virus now makes this antigen, and this virus is given, and therefore it becomes an antigen manufacturing factory inside the body, and it stimulates the immune system. And in all these cases, we are in the first 2, we are actually dealing with the actual pathogen either in a killed form or in a live attenuated form. But if you can simply take the gene coding for that particular antigen, put it in another organism like an E. coli or yeast, and if these microorganisms make these proteins in large amounts, this protein can be purified and given it as a vaccine. That's the recombinant protein based vaccines we're talking about. And more recently, we have what is called the nucleic acid vaccines, an example being the DNA or RNA, where instead of making the antigen elsewhere outside the body and then giving it, you make the antigen within our own body, either by giving as RNA or as a DNA, so that the antigen is made right inside our body, present to the immune system, so that you get either humoral or cell-mediated immune responses. All these vaccines are available in the SARS-CoV-2 vaccines, which I'm sure most of you, I don't think there is anybody in this audience who have not taken a SARS-CoV-2 vaccine. Everybody is taken a COVID vaccine. And the 2 most commonly used vaccines which are available in the country is the inactivated virus vaccine and of course the viral vector vaccine. I'm sure many of you must have taken either the Covishield or the Covaxin. Covaxin is nothing but an inactivated virus vaccine, where you infect cell lines with this virus and then grow the virus in large amounts, kill it and give it as a vaccine. In the case of the Covishield, a harmless virus called the adenovirus is taken, and the same surface protein gene of the SARS-CoV-2 spike protein gene is introduced into the adenovirus vector. So the adenovirus now makes large amount of protein, and this virus is actually given as a vaccine. Okay. So what is very interesting is that all these types of vaccines, India has been part of it. So we have to be very proud that all the, whether it's an inactivated vaccine or a live attenuated vaccine or viral vector vaccine, protein based vaccine or nucleic acid vaccine, an Indian company is involved. This is a very proud moment where all these 5 different major types of vaccines, Indian companies have been part of it. The other important component of a vaccine is what are called as an adjuvants. So in addition to the vaccine, you have what are called as an adjuvants, and adjuvants, primary purpose of an adjuvant is to modulate or enhance the immune response which are receiving the vaccines. I will not go into details. I'm sure there's an immunology course. You have been taught all these things. For a long time, the only vaccine that was available was the alum, but now you have several adjuvants that are now available. In fact, the vaccine which was not working very well, if you can change the adjuvant, it starts working much better. You can also modify the immune response of a vaccine antigen by using it with different kinds of adjuvants. So the adjuvant development has become a huge industry in the last 2 decades. So not only people are working on making vaccine antigens using these various platforms, people are also trying to develop novel adjuvants so that the vaccine efficacy can be improved by making novel adjuvants. So this has become a huge industry now. The recombinant hepatitis B vaccine that I'm going to talk about belongs to this category, where you take the gene coding for the surface protein of the hepatitis B virus, put them in yeast cells. Yeast cells now make this antigen in large amounts. You purify the antigen from the yeast cells and then mix it with alum, which is an adjuvant, and then give it as a vaccine. This is the recombinant hepatitis B vaccine. Now, the title of my talk says BEVAC, Kilovac B, COMBU5, and VAC-5. Now, what are these? BEVAC and Kilovac B are called monovalent vaccines because it has only one vaccine antigen, that is the hepatitis B virus surface antigen mixed with aluminum hydroxide or the alum. The COMBU5 and VAC-5 are called pentavalent vaccines because it has 5 vaccine antigens of 5 different pathogens. The hepatitis B surface antigen is present. In addition to it, it also has a diphtheria antigen, pertussis antigen, tetanus antigen, and Haemophilus B influenzae. So you take one vaccine, you get protection against 5 different diseases. So it's called a pentavalent vaccine. What is I am excited about is that the hepatitis B vaccine technology we are developing in my lab is a component of not only the monovalent vaccine, but also a component of the pentavalent vaccines. This technology was transferred way back in 2002, but still this strain is being used by the companies, and still these vaccines are being made. So the reason I keep talking about this technology is not that I'm talking about something that is irrelevant. A technology that was transferred almost 2 decades back is still relevant and still being used. Now, what is hepatitis B virus? Hepatitis B virus causes viral hepatitis. WHO estimates that almost 254 million people are still living with chronic hepatitis B. There is no cure for this infection. So the best way of preventing yourself from this viral hepatitis is to take this vaccine and prevent the virus from infecting the liver cells. And once it infects, very, very difficult to get rid of this virus, and you will become a chronic carrier of the disease and ultimately end up with a viral hepatitis and liver failure. So for example, in 2022, hepatitis B resulted in estimated 1.1 million deaths, mostly from liver cirrhosis as well as primary liver cancer or hepatocellular carcinoma. Best way of protecting yourself from this is to take these vaccines, about 3 doses from the childhood onwards. Now, how a hepatitis B vaccine was made, we have to go back to the 1980s, where when you go to the infected individuals, this vaccine, the virus, when it multiplies in the ears, the viral, what is called as a virus shedding, the empty virus particles are shed into the blood, and these are called as the DAN particles, and therefore people used to purify this shedded virus from the blood and then inactivate it and then give it as a vaccine. So this is called as the plasma derived hepatitis B vaccine. Although it was very successful, the problem was if you want to produce millions and millions of doses, it is not possible to go to infected individuals, take blood from them, purify the virus, kill it and give it as a vaccine. That is, scale up is impossible. It is just not possible. The other problem is with several bloodborne pathogens now becoming more and more important, you are always worried in the process of immunizing you against hepatitis B, you may end up with some other infection because it is infected with some other pathogen and maybe you are not purified properly. So eventually this plasma derived vaccine was discontinued in 1992. So today no plasma derived hepatitis B vaccine available anywhere in the market. It has been completely replaced by a recombinant hepatitis B vaccine. The biggest problem in developing a hepatitis B vaccine is, as I speak today, you cannot culture this virus. An important part of a vaccine development is that, especially if you want to develop inactivated virus vaccine, you should be able to culture the virus in large amounts. Only then you can immunize millions and millions of people. For example, the SARS-CoV-2 vaccine, the Covaxin became possible because you can infect a cell line called the vero cells with this virus, and therefore the vero cells in culture can now produce. You can grow this in large fermenters, so huge amount of virus particles can be made. You can then kill it and then give it as a vaccine. But if we cannot infect, if we cannot culture this virus in vitro, then scale up becomes impossible, and therefore, this became one of the major limitations for making a recombinant vaccine for, or a inactivated virus vaccine for hepatitis B. Now, if you look at the individuals who have taken these plasmid derived vaccines or who got infected with the hepatitis B virus, if I look at the antibodies that are being made in our body, almost 95% or 99% of the antibodies are made against the surface antigen of the virus. So, people thought if our body is making antibodies against the major surface protein of the virus, why can't, instead of giving the entire virus as a vaccine, why don't we simply give that particular surface protein as an antigen so that we can get protection against the disease by making this antibody. So, this antibodies made against the surface antigen will go and bind the surface antigen, preventing it from infecting the cells. So, this is the best way of protecting yourself. So, if you look at the surface of the virus, there are 3 types of surface protein called the L, M and S. The L protein is the one with the blue here there, and the green one is the middle antigen, and the orange one is the small antigen or the S antigen. And you can see 85% of the total surface of protein is comprises of the S antigen or the small antigen. So, most of the antibodies raised against our immune system against the virus is directed against the S antigen of the body. So, the challenge was, can we make this S antigen in large amounts? That is where the recombinant DNA technology came into help. So, you take the gene coding for the surface antigen of the hepatitis B virus, put in front of a yeast promoter in a yeast expression plasmid, put this expression plasmid inside the yeast cells, and you can culture these yeast cells in large amounts in huge fermenters, so that this antigen can be made in large amounts, and then you break open the yeast cells, purify this antigen by classic chromatographic techniques, mix it with aluminum hydroxide adjuvant, and give it as a vaccine. This is the process of making a recombinant hepatitis B vaccine. Why this vaccine became very successful is, when you make the surface pro-antigen, which is only about 15 kilodalton in size, after you lyse the cells and purify this antigen, this vaccine antigen self-assembles into a 300 nanometer particle called as the virus-like particle or VLPs. Therefore, it is highly immunogenic. So, this actually resembles the surface of a virus, and therefore, the antibodies raised against this recombinant VLPs actually can recognize the actual virus as well, and therefore, you get protection. Whereas, if you make the same antigen E. coli, it does not self-assemble into this particle, and therefore, it may be immunogenic, but it is not protected, because the antibodies raised against the E. coli produced antigen does not neutralize the actual virus, and therefore, it does not work. So, by luck or whatever it is, somehow the antigen made in yeast cells may form this VLP spontaneously, and as a result became a very, very successful vaccine. Now, the credit for making the recombinant hepatitis B vaccine goes to 2 companies, Merck and the GlaxoSmithKline or GSK. These vaccines, the Merck made a vaccine called as Recombivax, and GSK made a vaccine called Engerix B. Both became very successful and is responsible for almost getting rid of this disease in a preventive vaccine, as a preventive vaccine. So, these yeast-derived vaccines eliminated any concern for vaccine-related blood-borne pathogen transmission. You do not have to go to the infected individuals, you do not have to purify from plasma and all that. All these risks have been minimized. So, the hepatitis B vaccine is estimated to have prevented almost 38 million deaths over the lifetime of persons born between 2000 and 2030 in almost 98 low and middle income countries. So, this vaccine has made a huge difference in the quality of life in protecting, especially in developing people from developing countries. Now, if Merck and GSK is already making this vaccine, what was the need to make this vaccine in India? Why should you make it in India? The problem is, until 1998, the country was importing the hepatitis B vaccine, mostly Engerix B from GSK was coming. One vaccine used to cost, a monovalent single dose vaccine used to cost almost 600 to 700 rupees. There were also other brands, but 95% of the vaccine was actually Engerix B. And when government, all the developed countries started using the hepatitis B vaccine in their national immunization programs. So, India also said we should also introduce this vaccine, supplied free of cost to the citizens of the country. But if you look at the requirement, I am talking about the 90s, you require something like, oops, sorry, the pointer does not work. So, at that time, if you had to give 3 doses for about 25 million children, you require about 75 million doses of this vaccine. And if we have to import this many number of doses, the government will go bankrupt, because government just did not have that kind of money at that time. Remember, our economy was not that great as it is today. So it's too expensive to be included in the universal program of immunizations. So the government said, we have to make this vaccine cheap within our country, only then it can be included in the expanded program of immunization. So one of the programs started way back in the 1990s is a program called Technology Development Mission. So the government of India, especially the Ministry of, at that time it's called as MHRD, Ministry of Human Resource Development, now it is called Ministry of Education, asked the IITs and IISc said, why don't your scientists start interacting with some of the companies in India, and since the technology is available, why can't we make it indigenously, so that if more companies make this vaccine within the country, we don't have to import. Remember, at that time foreign exchange was also a huge problem. We didn't have that much foreign exchange, so we can not only save foreign exchange, vaccine also can become cheaper. Then only the government can think of introducing this vaccine in the universal immunization program. So they identified a number of mission mode projects, including a project in genetic engineering biotechnology. This is the at that time it was not called, today we call it as Make in India. Okay, this was thought of by the then government way back 2 decades ago as well. It had a budget of about 50 crores, and the idea is, once you develop a technology, whatever cost was involved in developing the technology, the industry which takes the technology should pay back the money. That should be the technology transfer fee. If I spent about 20 lakhs to develop this technology, if I transfer to industry, the industry should pay back the 20 lakhs so that the government gets the money back. That has been the philosophy. So we started this. As I said, I came back in 1993, and this was happening around 1998. A company called Shanta Biotech, the credit for developing the first indigenous recombinant hepatitis vaccine goes to Shanta Biotech. So they introduced the first indigenous recombinant hepatitis B vaccine in the country. Around that time, another company called Bharat Biotech, who had also just started the company, came back to me and said, can you help us to make a indigenous recombinant hepatitis B vaccine? So we signed a TDM project, and we did, as I said, Engerix B and the Recombivax was made in a yeast called Saccharomyces cerevisiae, but we decided to choose in a make this vaccine yeast called Pichia pastoris. Today the Pichia pastoris is called as Comagatella faffi. I will not go to details, but previously it used to call as Pichia pastoris. The advantage of this yeast over Saccharomyces cerevisiae is that I don't know how many people are taught glycolysis, Krebs cycle, metabolism. I think these days it's not fashionable to study metabolism, but I tell you, metabolism is very, very important. Saccharomyces cerevisiae is called as a respirofermentative yeast. If you give glucose to this organism, it generates energy primarily by glycolysis. And how many ATPs do you get from glycolysis? Can anybody tell? How many ATPs are generated from glycolysis? 2. Whereas if the organism completely metabolizes through Krebs cycle and oxidative phosphorylation, you get 38 ATPs from one molecule of glucose. You see which is more energy efficient. Saccharomyces cerevisiae primarily derives its, even though there is oxygen around, it doesn't completely oxidize glucose to carbon dioxide and water, therefore primarily generates energy by glycolysis, therefore that's why it doesn't go to very high cell densities, because there's not enough energy. Whereas the Pichia pastoris is called as a respiratory yeast, where it completely oxidizes glucose to carbon dioxide and water, therefore huge amount of ATPs are generated, very energy efficient, so there is surplus energy available, therefore it can activate all biosynthetic pathways, therefore it goes to very high cell densities. So if you inoculate a 1 liter of culture medium with Saccharomyces cerevisiae or Pichia pastoris, grow it for about 48 or 72 hours, you get much higher biomass from Pichia compared to Saccharomyces cerevisiae. That's what is shown here. So if I have to make a vaccine from 1 liter of a yeast culture, which organism I would choose? I would definitely choose a organism which gives higher biomass, because from 1 liter of the culture using the same raw materials, I'll be making lot more vaccine antigen than from Saccharomyces cerevisiae. So we decided, let us choose Pichia pastoris as our yeast host rather than Saccharomyces cerevisiae, which was being used by the GSK and Merck companies. The other important advantage of this Pichia pastoris is that it had how do you make a vaccine, how do you express an antigen in a yeast cell? You have to take the gene of your interest, put it in front of a promoter, right? Promoter is the region where the RNA polymerase and other transcription factors go and bind, and therefore the RNA is made and then it gets translated into protein. This particular organism has a gene called alcohol oxidase, and Pichia pesor is called as a methylotropic yeast. It can use methanol as a carbon source in addition to glucose and glycerol. Now, as long as you grow this organism in glucose or glycerol as a carbon source, this particular protein called alcohol oxidase is not expressed. You can see the band over there. But the moment you change the carbon source from glucose or glycerol to methanol, tons and tons of this protein are made, and you can see almost 30% of the total cellular protein is alcohol oxidase. So, it is one of the most powerful or very strong inducible eukaryotic promoters known, where as long as you grow the organism in glucose or glycerol, the promoter is not turned on. The moment you change the carbon source to methanol, huge amount of this enzyme is expressed. So, what do I do now? I simply replace the coding region of alcohol oxidase with the coding region of hepatitis B surface antigen. So, instead of making alcohol oxidase, now the yeast will start making my antigen of my vaccine antigen, namely the hepatitis B surface antigen. This is what happens. You can see we clone the hepatitis B surface antigen into the alcohol oxidase promoter, grow them to high cell densities. Methanol is not a very good carbon source, therefore, yeast cells does not grow to very high levels. So, you grow them using glucose or glycerol as a carbon source to high cell densities when the antigen is not expressed. Then you change the carbon source to methanol. Now, the promoter is turned on and huge amount of the antigen is made, and in about 4 different steps you can purify the antigen, and that also forms a nice virus-like particle as it happens in Saccharomyces cerevisiae. You now mix it with alum and give it as a vaccine. This is the technology. As I said, this technology was available elsewhere. All that we did is indigenization to make sure the technology actually works. It is similar to the metro coaches now being manufactured. Now, although the concept of metro coaches and subways are more than 100 years old, we are still struggling to make these coaches here in India. Indigenization is not easy. Although many of these things remain as trade secrets, trying to do something, we don't succeed. So, one of the important lessons to be learned is that developing countries have to put in a lot of effort to indigenize technology. For example, today you want to make a light combat aircraft. Technology has been there for 50 years with United States and Germany, but they won't give you the technology, right? And HAL is still struggling to make an LCA for the last 3 decades. So, many of these technologies are available elsewhere, but nobody is going to give you those technologies. So, we have to put in effort to indigenize these technologies because many of these have to come from everywhere. So, indigenization is a very important component for economic development of a developing country. So, we did this, developed this technology within 1 year's time, gave these technologies to Bharat Biotech. They made the grant was about 18 lakhs. So, the company paid about 18 lakhs to take the technology and they agreed to pay 1% royalty on total sales for about 5 years from the date of commercialization. Professor G. Padmanabhan, who was my mentor and also just retired as a director of IISc, contacted Dr. Abdul Kalam, who was the scientific advisor to the Prime Minister at that time, and said, this is an example of industry-academia collaboration. Those days, these are all not unheard. As Dr. Jyoti said, the job of an assistant professor in the Indian Institute of Science is to write projects, do academic research, publish good papers, train graduate students. This is the mandate, okay? Not to work with a company and develop some product. That's not the mandate. So, I took the road not taken, okay? And I suffered also, I'll tell you. It was not very pleasant. So, Abdul Kalam was told that this is an example of a good academia-industry collaboration and he should launch the product. The product was launched on October 22, 1998. The product was called Regevac B. But to my surprise, when I went to the production facility and took the vial, it said the vaccine antigen is being made from Saccharomyces cerevisiae. I got the shock of my life. So, I went to this guy, Krishna Ella, and asked him, "Hare yaar, why is Abdul Kalam here? Why is IISc director here? Why I am here if the vaccine antigen is being made Saccharomyces cerevisiae? Because I have nothing to do with Saccharomyces cerevisiae." He said, "Sorry, this is not your technology. Your technology will come later." This was the first disaster and disappointment in my life, where you work with a company for almost 1 year. You tell everybody that this is an example of industry-academia collaboration. You go to a product launch and you are told this is not your technology. This is our own. Later, I was told actually he was actually making it Pichia and selling it as Saccharomyces. Of course, you can't prove these things because these are powerful people. We waited for about 3 years. Technology was transferred in 1999. 2001, nothing happened. So, IISc served a legal notice. I am telling you all this because all of you think that research is do some research, put it on a fax machine, do some sorting, publish a paper, get a degree and go. Technology development is a different ball game, I can tell you. You have to be a jack of all trades. Science is only a small component of technology development, but ultimately it has to reach a common man. So many obstacles you have to cross. 2001, the IISc served a legal notice to the company. What the hell is happening? You have not, what have you done with the technology? Because it's an important technology. The country very badly needed it. And he responded to the legal notice saying that yes, blah, 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 we are going through all these procedures. We are now awaiting for inspection team to inspect the facility and give us approval for commercialization. As explained, we are yet to get a commercial license for manufacturing hepatitis B vaccine expressed in Pichia pasteuris. We at no point of time denied to pay royalty on the product, all that, but nothing happened. 1 year later, when I went to his website and found out, it says recombinant Revac B is now being produced genetic engineered yeast cells of Pichia pasteuris. So quietly he changed it from Saccharomyces cerevisiae to Pichia pasteuris, but never responded to any of our letters and was quietly making Pichia, and today he got Padma Bhushan for developing a SARS-CoV-2 vaccine. Well, this is part of the game. When you become successful, either by hook or crook, only the success remains visible to people, including the bosses or thing, but the mischiefs you have done in the process never comes to the light. So be it. So this was the disappointment that happened. So almost 3-4 years I slogged for this technology, but I did not get any credit. I was supposed to be promoted from assistant professor to associate professor. I had not printed enough paper, which is the primary mandate as an assistant professor. So our institute said, sorry, you cannot be promoted. So my promotion was denied from assistant to associate professor. So it took almost 2 years for me extra to get promotion later because I need to print papers. So the slugfest went on and all, but he became very powerful, so you can't touch them. He's the darling of the country who launched a recombinant vaccine and all that. So who cares for a assistant professor sitting in Institute of Science? I had no say. But this is the time there was a demand for recombinant hepatitis because one company or two companies making a vaccine is not going to bring the price down. Price goes down only when competition sets in, more players enter in the field. So many companies wanted this technology very badly. So Dr. Vijay Kumar Datla, who was the chairman and managing director of Biological Limited, came to me and then said, Professor, I still remember he came to Windsor Manor Hotel, called me and then said, Professor is actually making from Pichia only and selling it as Saccharomyces. We know you only gave the technology, please give the technology to us. Another company, Indian Immunological Limited, the executive director, Dr. Srinivasan, he also wrote me a letter saying that we also would like to make this technology because there is a huge market, many players. Even if you capture 2% of the vaccine market, it runs to crores of rupees. So they all wanted to make the hepatitis B vaccine. Biological Limited was actually making the DPT vaccine. This is the trivalent vaccine. But they said, if you don't mix hepatitis B, the company has no future. Because elsewhere, people are making combination vaccines. So if you don't combine hepatitis B with DPT, DPT is not going to sell anymore because hepatitis has become part of the combination vaccines. So they desperately wanted the technology. So it took at least 2 years for me to struggle with institute, convince my bosses that we have to give this technology to other companies because this guy is not going to give us because he is not even responding. So with great persuasion, the technology was transferred to these two companies in February 2002. Under the same terms and conditions, each company paid about 15 lakhs as technology transfer fee and agreed to pay royalty of about 1% of the total sales for 5 years. The first brightest moment of my career came on December 27, 2004, when I got this letter from Dr. Vijay Kumar Datla that this is to share the joyous moment of commercial launching of the hepatitis B vaccine produced with the clone developed by you. The product was formally launched on 22nd December 2004. My congratulations, blah, blah. This was the most enjoyable moment of my life after going through shit and going through so much of frustration, depression, and disappointment. This letter lit me up. So I really started jumping up and down, saying that yes, finally something came out of my work. Same thing happened. Indian Immunologicals took 2 more years to launch the product because you had to do limited clinical trials. They sent me a letter on 29 September saying that this product has been launched by Honorable Minister of Finance and Planning, Government of Andhra Pradesh, Rosaya. Sorry, I decided not to go for these product launches because I had a harrowing experience with Bharat Biotech. You go there and finally the company says, this is not your technology. I said, I'm not coming for any of your product launches unless you give me a letter. So they both gave me a letter saying that, yes, this is your technology and we're happy. The companies, unlike the golden star or golden boy of biotechnology today who cheated me, these guys promptly paid. Every next 5 years, they paid the royalties. They were selling about 1 to 2 crores of the vaccine every year. Market had still not picked up. Remember, the market picks up only if the government starts buying in bulk. Still, the price was high. Government was just waiting for more players to come in so that more vaccine is available in the market, so the price comes down. So the government was still not ready to make large purchase of this vaccine to introduce into universal program. So they were not selling. They were only selling for small amounts. Indian Biologicals also said between the 2000, because the MOE lifespan was about 7 years, so the MOE expired in 2009. So till 2009, they sold about 3 crores and paid 1% royalty. The problem is the market picked up after the MOE expired. Another lesson I have learned, it is not enough if you are a good scientist, you should be a good, like your pro vice chancellor, you should be a good finance guy. You should have the forethought. Products in biology takes time to pick up, market, develop, go through clinical trials, get into the market, compete. It takes time. It is not something that today transfer technology, tomorrow you start making money. This was not, nobody told me all these things. So we said the MOE is valid for 7 years, but the market picked it up only after the MOE expired. So the government started floating huge tenders. Remember, if you want to introduce the vaccine mass immunization, you require millions of doses. Each tender would be for 100 million, 200 million doses. And also it became, this is the time the combination vaccines became prominent across the globe. So giving just, if I had to give vaccination for 4 diseases, I cannot ask the child to come for 4 times and prick 4 times. So that concept is gone. So they were combining vaccines. So both these companies were forced to combine hepatitis B with other vaccines. So pentavalent vaccines became the gold standard across the globe. So Biologically Limited launched the combination vaccine called as COMBE5. So it contains hepatitis B, diphtheria, pertussis, tetanus, and Hib. So the government started floating tenders for pentavalent vaccines. The hepatitis B vaccine component was still coming from my strain. The government of India launched, this is the first NDA government that came in 2014. 2015, they launched this program called Mission Indradhanush to freely supply vaccines across the population, first in a limited number of states and then spread it across, extend it to the entire country. And in March 2021, I saw this news item that Biologically Limited gets an 895 crore order for the supply of liquid pentavalent vaccine. Okay, you see the market size. One government order is 900 crores. That's the kind of need for the vaccine. So people who are making this vaccine apply for, give their tenders, the lowest tender gets picked up and he starts supplying the vaccine. So Biologically became the successful bidder for this vaccine and they got this huge order to supply liquid pentavalent vaccine for the government of India. So I saw this news item. Remember, the academic scientist goes into the background once you transfer the technology. Very rarely companies tell you what is going on because you become irrelevant after some time. So after seeing the news item, I thought, what the hell, yeah? I mean, you got a huge order. And I wrote to the company, can you pay some royalty, 895 crores, 1% royalty is bloody 8.9 crores. They said, sorry, your MOE has expired. We cannot give you any royalty. Then I said, at least, are you still using my strain? Can you at least give me a letter saying that you are still using my strain? By the time Vijay Kumar Datla died, his daughter took over as the director. She very promptly sent me a letter saying that we are still using the strain provided by IISc for hepatitis B vaccine production. And since 2010-11, we have sold more than 110 million doses of hepatitis B vaccine as expanded program of immunization as our BEVAC brand, that is the monovalent vaccine. And we have also sold more than 200 million doses of the hepatitis B vaccine as the pentavalent vaccine. And BE acknowledges the vital role played by IISc for our vaccine development program. So again, I started jumping up and down that here is a technology that is still was being used even after almost 15 years. Around the time, Indian Biologicals also realized the market potential, realized there is scope for multiple players in the market. They also developed a pentavalent vaccine called Vaxstar 5. By the time again the person with whom I interacted had retired, a new guy had taken over called Anand Kumar. So again I wrote to him, "Hey, I gave you the strain, are you still using it?" Again, they promptly sent me a letter. They also, once the BE order for vaccine supply for 3 years got over, government forwarded new tender. Immunologicals bagged the tender. It is just my luck. The two companies with whom I interacted, both of them bagged the government tender. They got a 210 crore order for government to supply vaccine for 2 years. So again, it was good enough. Rajendra, the person with whom I worked for recombinant vaccine development, he was the product manager. They again promptly sent me a letter saying that we're happy to inform you that the Pichia strain received from IISc has been used to make our monovalent and pentavalent vaccines, and this many doses have been made, and we acknowledge the vital role played by IISc. So you can see the reason I'm sharing is not to just beat myself. This is to just tell you, many of you, some of you may become entrepreneurs tomorrow, develop products or process and contribute. But it's not an easy journey. No, much more easier for you to do some research, publish a paper, other journal, and then look at its impact factor, and then go abroad and do some postdoc. It's a much easier path. But to develop a product, work with a company, and finally get credit for your work happens very rarely. And luckily in my career, it happened, although I went through a lot of tough time initially. Again, lesson I tend is that don't give up. Yes, everybody goes through ups and downs, disappointments and depression. That's part of your career. But you need to keep going and find out that everybody is not a crook. There are honest people around. It's just bad luck. It happened. You had a bad experience. But just keep moving. Just about a year back, Indian Institute of Science conducts alumni meetings, alumni association meetings, where they invite their alumni to, they organize a nice conference every year, usually around December. 2023 December, IISc said, why don't you talk about your vaccine work? The title was Vaccines from Discovery to Market and Their Journey. You also put a product in the market, why don't you share your experience? I told them I will talk only if the company says we are still using your strain. If the technology had become irrelevant, I'm not going to talk about it. So I again wrote to the company, Dr. Mahima Datla on 25th November 2023. Hope you will recall that IISc had transferred the Pichia strains which are being used. I would very much appreciate if you can provide an update on how many doses of BE-Vac and COMB have been manufactured till 2023. She passed on to a guy called Vikram Paradkar. Next day, the guy replied to me saying that, yes, 850 million doses of COMB has been made so far, and 225 million doses have been made for the monovalent vaccine. I got a similar letter from Indian Biologicals Limited saying that we have made about 144 million doses of Filovac B and 168 million doses of the pentavalent vaccine. So this was very heartening because these days, considering the example I gave you in the beginning, the guy who just ignored me and completely ditched me, there are companies which remain truthful, which remain honest. Not only they pay royalties, but they still appreciated the technology even 2 decades after the technology transfer. This is very, very heartening. I thought I should share these examples because this road, if any of you decide to take this road of an industry, environment, and developing products or projects, it's not an easy road. It's full of obstacles and disappointments and depression, but you need to keep going, hoping that something good will happen. It's not an easy road to journey. This is a summary of the story that I want to tell you. Technology was transferred to 2 companies, 3 companies actually. One company I don't talk about because the company never acknowledged my contribution. Both the companies, technology got in February 2002. The monovalent vaccine was launched in 2004 and 2006, and so far 225 million and 140 million doses have been made as of November 2023. The pentavalent vaccines were launched in 2011 and 2018, and BE has made about 850 million doses of COMB-EFI. Indian Biologicals made about 169 million doses. So about 1,400 million doses of the vaccine have been made so far, not only for within the government India, the vaccine also being exported to a number of developing countries, okay, across the globe. So it's a very, I feel very happy about it. Those of you can just do a Google search, IISc hepatitis B vaccine. If you do a Google search, you get this article which was published in a Connect magazine. I have shared my experience of the hepatitis B vaccine journey, how it all went through. You can read more about it. Is this technology still valid? Hepatitis B vaccine is going to be here for another 3 decades because it is still a problem. So there's a huge market. You can see year by year the market is only increasing. So these vaccines are here to stay. Okay. So companies will continue to make this vaccine and hopefully they will not replace my strain, because if you bring a new strain, you have to start clinical trials all over again. So companies, unless they find there is another strain which is making 5 times more antigen than my strain, they would not like to change any of this, because if you change a vaccine strain, you have to go through clinical trials again. So companies usually don't want to change these things unless they find a huge commercial benefit. They don't want to change the technologies. Newer vaccines are coming. For example, in the 1990s, instead of using yeast cells for making the vaccine, a mammalian cell derived vaccine is available, where they have included not only the S antigen, they have also included the pre-S1 and pre-S2. This vaccine is now being available, but all those studies suggested that these vaccines are more effective. Remember, the yeast derived vaccine, there are about 10 to 15% of the people are called non-responders. If you give this vaccine, you don't develop antibodies. For some reason, they are called non-responders. So you think that they have taken a vaccine, but their body doesn't make antibodies. And these are all based on data. Still, we do not know in our country what is the percentage of non-responders among the Indian population. I don't think data is available. This is another problem. We are very poor in epidemiological research. All the data is available only from the Western countries. Whether the same thing is true for India is not clear. So there are 10 to 15% of the people who don't develop antibodies. They're called non-responders. So we now take this mammalian cell-derived vaccine, which also has pre-S1 and S2, even these non-responders start responding. So there is an advantage. 2017, another new vaccine has come, where called as Heplisar-B, the yeast-derived recombinant hepatitis B was combined with a novel adjuvant called cytidine phosphate guanosine nucleotide that binds the toll-like receptor 9 and simulates a more robust immune response. I told you, adjuvant manufacturers have become a huge industry now. So instead of aluminum hydroxide, if you put this adjuvant, you get a much better immune response. So this became a newer vaccine now. 2021, another vaccine has come called Pre-Hebrio. It's a tri-antigenic HB vaccine, contains pre-S1, pre-S2 and S, but it's been approved only for adults of 18 years of age and older, not recommended for pregnant persons or those on dialysis, because they haven't done clinical trials in these patients. So newer vaccines are still coming up, but a conventional yeast-derived vaccine is still, by and large, is the 95% of the market is still captured by the conventional yeast-derived vaccine. Okay, what next? Okay, I have told you a story which is 20-year-old. I'm simply going around talking about my past all the time, and I have known something else the last 20 years. Our lab has been, vaccine development is only a small, not the main job. As I said, as a professor or assistant professor at University of Sam, my job is to do basic research, train graduate students and publish papers. These are the various research programs that we have in the lab. We have a big program on understanding the gene regulation in picopastories. The reason why I got it, because as I told you, the promoter that is being used for making this vaccine is called alcohol oxidase promoter, which gets induced when you put methanol as a carbon source, but not glucose or glycerol. Now, as a person who has been working in the area of gene regulation, I asked the question, why is this promoter did not or is not turned on when the carbon source is glucose or glycerol? Now, two things must be happening. How is transcription activated in eukaryotic cells? Does anybody tell you? How is a gene turned on or turned off in eukaryotes? Have you people, do they study eukaryotic gene regulation? So usually promoters have binding sites for what are called the transcription factors. When transcription factors go and bind to the promoter region of a gene, they turn on the gene. When transcriptional repressors go and bind, it shuts off the gene. So what should be happening? There must be a transcription repressor that is going and binding to the promoter of alcohol oxidase gene, which prevents its expression from in glucose or glycerol. When you change the carbon source to methanol, the repressor may be coming off and an activator go and binds, therefore it should be turning on. Nothing was known when we started. So although companies have been using this promoter for making hepatitis B vaccine, no basic research was being done to understand how is this promoter turned on or turned off using methanol or glycerol as a carbon source. So we initiated a new program to understand are there transcription factors that are binding to this promoter. We have contributed to the discovery of 3 transcription factors called MXR1, ROP1 and TRM1. I will not have time to go through details. This is one basic program that has been going on. More recently, we have exactly identified what sequence to which a transcription factor binds. As you know, transcription factors are sequence-specific DNA binding proteins. A transcription factor contains 2 domains, a DNA binding domain, a transcription activation domain. Through the DNA binding domain, it binds to a very specific sequence in the promoter region. through the transcription activation domain, it interacts with RNA polymerase and other proteins and activates transcription. So have mapped the exact sequence in the alcohol oxidase promoter to which the MXR1 transcription factor binds, and only when it binds, methanol can activate the expression of this gene. If you mutate the MXR1 binding site in the promoter, methanol cannot activate the transcription of the gene. All this has been published. More recently, we have identified a 9 amino acid sequence that serves as a transcription activation domain. If you simply take a 9 amino acid sequence in this 1155 amino acid protein, this 9 amino acid sequence, if you attach to a DNA binding domain, it can activate transcription. This work is going to be published in the March issue of JBC. We are also now studying what is called the glutamate metabolism. Pichia is again a very unique yeast because it can use amino acids as a carbon source. Saccharomyces uses amino acids only as a nitrogen source. Again, I will not go into the details of metabolism. I don't know how these days students are only interested in molecular biology, recombinant DNA. Nobody wants to study classical metabolism biochemistry, so I will not go into details. But metabolism is very important. I will tell you how understanding the metabolism by which this yeast utilizes amino acids like glutamate as a carbon source has now led to the development of a new expression system in our lab. We also have a new program called understanding host virus interactions. When a virus infects our body, many genes in the host cells are turned on. Our body turns on many genes. Some of these, like for example interferon responsive genes, they are involved in antiviral response. There are also some genes which are activated which actually helps the virus to multiply better. So there is a proviral, there is an antiviral response. Both these genes are activated when a virus infects our cells. So at that time in the 1990s, we started a research program to understand when a Japanese encephalitis virus and rabies virus infect our cells, what kind of gene expression changes takes place inside our cells. And we have identified many genes which are activated when the virus infects the cell, and one of such important genes is called as virus inducible non-coding RNA. This is a nuclear non-coding RNA which is activated when JEV or rabies virus infects the cells. And I will not again go into the details. This gene has 5 years later, this gene was renamed as NEAT1, nuclear enriched abundant transcript 1, and now they have shown this gene, this particular RNA is not only made during virus infections, it is also made when normal cell becomes a cancerous cell. That is, when the normal cell turns cancerous cells, this gene is upregulated, and this gene has a role in anti-proliferative responses. So now, if you now go to a company like a MedGenome, or if somebody has got a cancer, and you take the cancer tumor and do a genome analysis or transcriptome analysis, if they find the NEAT1 or WNK is upregulated in your tumor, then it becomes a good prognostic marker. That means you are likely to respond better to a treatment. So upregulation of the WNK or NEAT1 in cancer cells has now become a new prognostic marker for cancer. We have also, in collaboration with Professor G. Padmanabhan, we have also been contributing to development the malaria parasite biology. I will not go into the details. I will just take about 5-10 minutes to tell you some of the, just to tell you why biochemistry metabolism is important. All this, not only you can do basic research, you can also do a lot of applications. Some of our students are not motivated to study glycolysis, Krebs cycle, amino acid metabolism, but these are all being forgotten now, but they are all very important. As I told you, this particular yeast not only uses glucose, glycerol, or methanol as a carbon source, it can also use amino acids. The normal common medium that people use for growing yeast cells contains yeast extract, peptone, and a carbon source called YPD. Yeast extract, peptone, glucose, or glycerol as a carbon source. Peptone contains amino acids, basically. It's a peptide, trypsin digest of a peptone. This particular yeast, you can just grow it in yeast extract and peptone. You don't have to add a carbon source. Because it has an enzyme called glutamate dehydrogenase, which can convert glutamate to alpha-ketoglutarate, and as you know, alpha-ketoglutarate is a TCA cycle intermediate. So the glutamate can enter TCA cycle through this enzyme, and therefore it can generate energy. And also there is another important enzyme. Whenever you grow a yeast or any cells, a carbon source other than glucose, it has to activate a pathway called gluconeogenesis. That is, it has to reverse glycolysis has to take place so that the pentose phosphate pathway is activated, ribose sugars have to be generated, without which DNA cannot replicate. So whenever you grow an organism, including humans, if you consume a carbon source other than glucose, a pathway called gluconeogenesis is activated, and a key enzyme of the gluconeogenesis pathway is called phosphoenol pyruvate carboxykinase or PepsiK. So when I grow this yeast in a amino acid is a carbon source, as you can see, as long as you grow this yeast in glucose, glycerol, ethanol or acetate as a carbon source, this is the western blot, glutamate dehydrogenase is not synthesized. The moment you change the carbons to glutamate dehydrogenase expression is induced. Similarly, PepsiK is hardly expressed when you grow in glucose, because you don't require glucogenesis when you grow in glucose. You don't have to make glucose. So PepsiK is turned off. But the moment you switch to other carbons, especially when you put glutamate, huge amounts of this protein is made. This is the first time we are showing that an amino acid like glutamate can induce the express of two proteins. There is no example so far in the literature that an amino acid can induce the synthesis of two enzymes required for its metabolism. So we showed it, that means a glutamate inducible synthesis of two genes we have identified. If you now replace the coding region of PepsiK or GDH with GFP, now GFP is also synthesized in a glutamate inducible manner. GFP is not made when glucose, glycerol, ethanol or acetate, but the moment a carbon source glutamate, huge amounts of protein is induced. This basic research, this idea that you can turn on the expression of two promoters by putting glutamate, has led me to think, why can't I develop a glutamate inducible expression system? Just like there is a methanol inducible expression system using alcohol oxidase, why can't I develop glutamate inducible expression system using glutamate as a carbon source? So we actually filed a patent on this and it's been just published last year, where we have developed a glutamate inducible expression system, and using SARS-CoV-2 protein as a model protein, we actually demonstrated that if you now clone the receptor binding domain of SARS-CoV-2 under the methanol inducible alcohol oxidase promoter or the glutamate inducible PepsiK promoter, you can actually make a lot more of the vaccine antigen through our promoter than the methanol inducible promoter. That means we have now developed a novel expression system where instead of adding methanol, you can add a glutamate as a carbon source. What is the advantage of glutamate? We've actually showed that you don't actually require glutamate, you can simply use monosodium glutamate. I'm sure all of you who are eating Chinese dishes, they keep sprinkling that salt whenever they make noodles or something. That's nothing but MSG, okay, which is a FDA approved food additive which gives the taste called as Ajinomoto, very popular name. Available in tons, 100 grams of Ajinomoto is 40 rupees. You can order from Amazon, very cheap, the water soluble, available in tons. So we thought, here is an example where monosodium glutamate can be used as an inducer for making your recombinant protein of your interest. This was just published. We got an Indian patent recently, and the data is also available in the public. Indian Institute of Science decided to publicize it so that the industries are aware of what is going on. So basically this is what we are saying. Instead of cloning your gene of interest under the alcohol oxidase promoter, which is methanol inducible, you clone the gene under PepsiK promoter. You grow the cells in large amounts in glycerol or glucose, a carbon source, when PepsiK promoter is not expressed. So it generates sufficient biomass. Now you then change the medium to MSG. Now the recombinant protein is made, and you can now purify this and use it as for whatever purpose. This was also publicized in Times of India, saying that IISc finds a safer way to mass produce recombinant proteins like insulin using a novel, non-toxic. Methanol has a lot of problems. Methanol is a toxic molecule. The first reaction, conversion of methanol to formaldehyde, which is what alcohol oxidase does, generates hydrogen peroxide. As you know, hydrogen peroxide is very toxic to cells. So cells become sick, and it also generates a lot of heat. And storing methanol is a problem because it's a hazard molecule. You need to get explosive license and all that. So people have been looking for alternative expression systems in PICIA. So our system, where we say instead of methanol, you can use MSG, is now gaining attention. This is a recent, just in January, now that all these newspapers have published, this has now caught the attention of some of the industry. Some of the industries are now coming and asking the question. This is a recent email I got. I don't want to talk about the name of the company because discussion is still at the preliminary stage. Whether we have been working with PICA Pastores, employing PICA-based technology for an insulin portfolio. We want to connect with you, discuss the recent advance in this area, and explore any possibilities of working to develop a high expression platform. So right now, insulin is being made in PKI using the methyl-inducible alcohol-lysis platform. Now, if we can, the same strain, if we can now also introduce another expression cassette where the insulin is being made from a pepsi-K promoter, you can now express both from methanol as well as monosodium glutamate. So, insulin yield can go up because a lot more players are now entering the insulin market. So, using the existing infrastructure, if you can improve the yield of, if you can develop a new strain which makes more insulin, we can be more effective in competing with others. So, companies are now interested. So, this is what I am saying. Right now, the companies are using alcohol-oxidase promoter for making insulin. Now, I am saying that you can also making insulin if you clone the gene under pepsi-K promoter. So, if you now add both methanol and MSG, can you double the vaccine antigen levels? Or, of course, everything depends on system doesn't work the way we think. We don't know whether the yield will improve 2-fold, 3-fold, 4-fold, we do not know. But if the yield goes up and if the company thinks it is commercially viable to change the vaccine strain from just the methyl-inducible strain to methanol for MSG, then it can become a commercially viable strain. We have also, as I said, not all your basic research, when you try to translate, becomes successful. There have been examples where we have not been able to successfully launch a product in the market. We have also been working on developing a DNA vaccine for Japanese encephalitis and rabies virus. We have published, we have even filed a patent, but unfortunately we could not bring a product into the market. 2 students, PhD students contributed to this work. Ashok contributed for the demonstrated a DNA vaccine worked very nicely against Japanese encephalitis in a mutant intracerebral challenge model. Shubhabrata Biswas actually demonstrated a DNA vaccine for rabies works in mice, cattle, as well as in dogs. This was done with Indian Immunological tie-up. These are all the publications that came out. We even filed a patent on this. The Department of Biotechnology, when Atal Bihari Vajpayee was the Prime Minister, he coined a new term, Jai Jawan, Jai Kisan is the old one. They also added Jai Vigyan. Jai Jawan, Jai Kisan, Jai Vigyan. Because he said knowledge is also very important. So, the Department of Biotechnology started a program called Jai Vigyan Vaccine Mission Program. So, develop a mission mode projects, new vaccines. So, DBT funded for preclinical, as I said, if you want to put a biological to the drug or a biological to the market, you have to do what are called as preclinical toxic studies. You have to first demonstrate that your product is not toxic in animal models. You have to test it in 2 animal species to show that your product is not toxic. Then you have to do what is called a phase 1, phase 2, phase 3 human trials. Only then you will get approval for using in humans. All these things takes anywhere from 2 to 10 years. Okay, that's not easy. So, we did the preclinical toxic studies at National Institute of Nutrition. Again, all these were published. The company even said we will first launch a veterinary vaccine called Dinarab. But as I said, the person with whom I was working, Dr. Srinivasan, retired. New person came in and he said, this is all rubbish, I am not interested. He had his own vision to take the company forward. He said, this is not going to make huge amount of money for me, so I am not interested. And this is what happened. Just got buried. Okay. So, as I said, this is a path that's full of thorns, not a smooth road. You spend a lot of effort and time. You can publish papers, but whether it can really become a product, the successful product in the market, and whether the company will acknowledge your contribution is a big question mark still. So, it doesn't happen all the time. This is the most recent paper from our lab, just it's going to come out in March issue of Journal of Biological Chemistry. Again, IISc has put up on the website, publicizing it, saying that's a very important thing where we have shown a 9 amino acid sequence. So, this is the protein called MXR1, which is 1155 amino acids. In that, as in the orange one, you have a 9 amino acid sequence. And if you now take this 9 amino acid sequence, put in front of the DNA binding domain, it now binds to the MXR response element in the target promoter region, interacts with what are called a histone acetyltransferase called GCN5, removes the histones and activates transcription. So, tiny but mighty, just 9 amino acids, but it's very mighty. It can function as a transcription activation domain. This is briefly the thing. MXR1 is a huge protein called 1150 amino acids. Using an algorithm developed by a US lab, we have demonstrated that there are at least 22 9 amino acid sequence that has the potential to function as transcription activation domain. And we in the 2021 we actually demonstrated that this sequence called QELESSLNA 9 amino acids can function as a transactivation domain. If you delete this 9 amino acid sequence, MXR1P cannot function as a transcription factor. Although it goes and binds to the MXR1 alcohol oxidase promoter, it cannot activate transcription, saying that this sequence is required for activating transcription. We also identified there are at least 20 other such potential sequences which can function as transcription activation domain. Now, but how do we experimentally validate it? This is what we have done recently. We have taken just the DNA binding domain of MXR1, which contains 2 zinc fingers. We made a synthetic gene in which 3 copies of this 9 amino acid transfer activation is attached to the DNA binding domain, and we made the synthetic transcription factor and added an amnion green, which is like a GFP. You can visualize it very clearly, or a histag to visualize that the protein is actually getting expressed in the cells. This is the alpha fold structure of this synthetic transcription factor. These 2 transactive domains form an independent fold, and when you transfect into cells, they go nicely localized into nucleus. Now, for a protein to function as a transcription factor, it has to go into the nucleus. Now, if it stays in cytoplasm, it cannot activate transcription. We showed that this synthetic transcription factor can actually go into nucleus and act nucleus, and once it goes into the nucleus, this transactivation domain interacts with GCN5, which is a histone acetyl transferase, and activate transcription. When you delete GCN5, it does not activate transcription anymore, indicating that these transcription factors activate transcription by recruiting an histone acetyl transferase to the promoter, remove the histones in the promoter region so that RNA polymerase can go and bind and activate transcription. So, using this synthetic transcription factor approach, we made 21 synthetic genes in which each synthetic gene has a DNA binding domain linked to 3 copies of each of these putative 9 amino acid TADs, and shown that 10 out of these 21 TADs actually function as a transactivation domain. This is the most recent study. I think I'll stop here. All this work was primarily done by PhD students in my lab. About these are the number of PhDs who have contributed. The work on hepatitis B vaccine was actually done by Anshu Bhatnagar and started off, and the glutamate metabolism work was done by Trishna, Krishna Rao, Sminara. These are the current students. Neetu, I have not talked about her work. She is actually trying to understand how glutamate dehydrogenase and PepsiK synthesis takes place. There is a basic helix loop helix protein called RTG1 that actually plays a very important role in inducing glutamate inducible synthesis of PepsiK. She is trying to understand the basic aspects of this. Prachi is the first author in the JBC paper that's going to come out in March. These are the 2 project assistants who helped in the JBC, the microbial cell factors paper that came out last year, and she is the current one working with Neetu. And all these papers, and Aditi is the one who identified the 9 amino acid transactivation domain. And I thank all these funding agencies for, without money you can't do any research. Money is very important, and I thank them all. Thank you very much.
Thank you, Professor Rangarajan, for that really enlightening talk, from going from how academia industry collaboration can sometimes not go as you expected, but then to that success story with the other 2 companies. I'm sure the students have a lot of questions. We'll be happy to open the floor for questions, please. If someone doesn't have yet or still thinking about it, maybe I will start by asking about the adjuvants. I realize that that's quite something, I think in basic research, especially for the immunology students, that we should be also in India thinking about. Do you have any thoughts?
I don't think any new adjuvant has
come
out. Like Bharat Biotech vaccine actually uses a new adjuvant, which stimulates your response. So if you now simply type Covaxin, they have used, I think it's monophosphoryl-lipid or something like that. It's a novel adjuvant which stimulates TLR2 response. So, but these adjuvants are all coming from elsewhere. I don't think anybody has developed any new adjuvant within the country.
So there is a scope for indigenous
adjuvant. If you develop a new adjuvant today, you can become a millionaire.
So I think this is for the young entrepreneurs. That's something definitely, you know, it should maybe pique your interest to try and think about that. Yeah.
Hello, sir. I'm Vinay here. So you said for a vaccine to come into the market, it would take anywhere between 2 to 10 years. So I just wanted to get your thoughts on this COVID vaccine which came so early into the market. So what are your thoughts on that? Like,
COVID vaccine is a kind of a thing, because they all granted what is called an emergency approvals. It's called emergency user authorization or something like that. Because the pandemic is there and they very badly needed, so they broke all the rules and then gave the approvals. If you now read, there is a huge controversy going on in US, for example, even the mRNA vaccines that they developed by Pfizer and BioNTech. They are now saying that it has been rushed into the market. The mRNA vaccines have a lot of side effects. There is what is called a long COVID. People suffer from long COVIDs. It affects your heart function. People are coming up with neurological disorders. There is a lot of controversy going on. There is a pro-vaccine lobby. There is an anti-vaccine lobby. You don't know what is the truth. But these vaccines came in a market within 6 months because they were granted emergency use authorizations, which normally does not happen. Even for Covaxin, even before the preclinical toxic studies got published for Covaxin, approval was given for starting phase 1 human trials. There was a lot of push at that time. Like our DNA vaccine never came to the doctor because the DCGI, the Drug Control General, never gave us approval for starting a phase 1 human trial, because we have what is called a Drug and Cosmetics Act. According to the Drug and Cosmetic Act, those days, I'm talking about the 2000, if a vaccine or a drug has not been approved for a phase 1 trial else outside India, you cannot start a trial in India. This was the Drug and Cosmetic Act. So the Drug Control General never gave, because no DNA vaccine was in the market at that time. So although we completed the preclinical toxic studies, the DCGI did not give us permission for starting a phase 1 human clinical trial, because no DNA vaccine was approved in the market. Okay, we studied, struggled, all kinds of strings were pulled, but nothing happened. Okay, so that's why we had to give up. Okay, so normally it takes a lot of time to do these trials, but COVID vaccine is a different ballgame because there was a need for immediately immunizing the population. These were given emergency use authorizations, bypassing all the regulatory procedures.
Yeah.
Thank you, sir, for such a wonderful talk. I have basic questions, and does it make sense? Does this antigen requires a post-translational modification to have a immune response?
Which one?
When what you got from the HV, because in E. coli,
Hepatitis B, no.
Yeah,
Doesn't
because E. coli it was not as successful, but in
E. coli was not successful because it was not forming the VLP. That's why it was not successful. For it to become immunogenic, it has to form this VLP. Only then the antibodies raised against the E. coli made protein does not recognize the antigen present on the virus surface, so it does not neutralize. For your vaccine to work, it has to generate virus neutralizing antibodies. So the VLP, the antibodies made against the VLP that is made in yeast can also bind to the actual virus. Because the surface of the E. coli antigen does not generate virus neutralizing antibodies, so it became a failure.
My question is regarding the JV vaccine. Why do you think it didn't take up? Is it because of the fact that JV sort of like stays in the brain and it's...
No, this has nothing to do with science. It's all paisa.
Okay.
As I said, to do preclinical trials, clinical trials, it's a very expensive and long process. Unless the company says, company is not there for charity, they have to make profit. Unless the company says that there is a market for this vaccine, I can make money out of it, they are not willing to come and spend money and take your technology to make vaccines. JV had a very, whom do you want to immunize? What is the target population for JV vaccine? Not clear.
Yeah.
Hepatitis B vaccine, the moment you are born, day 3 you are given a shot, right? For JV, you don't know what is the target population. Whenever there is an epidemic, the minister will announce, Kolar, there is a JV epidemic, go and give vaccine. But will you take a JV vaccine? You are probably here. The chance of you getting a JV is very remote.
Yeah.
So there is no market for a JV vaccine is not clear. So it's economics supersedes science.
Okay.
You can do good science, but if economics is not good, it will not take off.
Thanks.
Any questions from the student?
Hi.
Yeah.
There, yes, yeah, please.
Hi, sir. Great talk. So I was wondering about the methanol-induced activation of the promoter. So if you say it's activating the promoter, also do you think, and have you wondered about the growth rate or the survival of the organism as it progresses?
See, as I said, methanol is not a very good, there are only 2 carbon atoms in methanol, okay? The organism spends a lot more energy to cleave the methanol and bring it to the Krebs cycle. More energy is spent on bringing it to Krebs cycle than actually generating energy from Krebs cycle. So it's not a good, that is, you are not generating sufficient energy, therefore it's a poor carbon source. That's why you have to grow the organism first in glucose or glycerol to generate enough biomass. Then you change. So in this biomass, you change the carbon source, that whatever the existing biomass now starts producing antigen. So there are what are called as good carbon sources, there are what are called as poor carbon sources. Amino acids, methanol, ethanol, they are not good carbon sources. Because organism spends a lot of money to make glucose from these carbon sources. Gluconeogenesis is an expensive process. Whereas if you use glucose immediately under pentose phosphate pathway, glucose 6-phosphate goes into pentose phosphate pathway. But we use methanol, acetate, ethanol, amino acids through the Pepsi K, it has to now go back, reverse glycolysis. All these things generate.
No idea about
it's approved by US FDA.
Okay.
The carcinogen, US FDA will not approve it. Okay. There are controversies, there are a lot of literature saying that there is no problem. Only problem is it's a salt, no? It's basically you are consuming more salt, because to addict you to a particular taste, you go to a Chinese restaurant, you see that white powder is sprinkles on your noodles, it's all MSG. It's called umami, it's a taste enhancer, no? So you get addicted to the taste. Therefore, in the process you are consuming more salt, and then your BP will go up. That's the problem, okay? So salt consumption is not good for health. But my system, you are not going to consume MSG. MSG is just going to induce your synthesis, and it is the organism which is consuming MSG. You are only interested in the protein that is being made. So you are not going to consume.
Yeah, please go ahead.
Hi sir, it was a great talk. I just have a doubt about the amino acids that you said are acting as a transcription activation domain. So is it conserved between genes? Has it been looked into, or is it different for each gene, something like that?
No, there is an algorithm, okay? You just go to the site, you just do a Google search on 9 amino acid transactivation domain. It takes to the lab, so you feed in any amino acid, any protein sequence to that. Using the algorithm, it actually predicts what 9 amino acid sequence this protein can have the potential to form as a transactivation domain. This was developed by, I forgot his name, it's a lab in US which has developed this algorithm. So you can take any protein sequence and simply go to the website and feed it. It gives you. It gives you at low stringency, moderate stringency, high stringency. Like if I go to high stringency, it says 0. We actually predicted this 21 potential sequences using moderate stringency. Ultimately, when I do the extraction, only 10 of them actually worked. So it's a prediction. It's a prediction tool available, so you can test. But there is no guarantee that every prediction comes true. Only some of them. But they have done a lot of work to predict that this particular 9 amino acid has the potential to function as a transactivation domain. If you now just do a Google search, there are about 90 transcription factors in which they have identified this. Just do a Google search 9 amino acid transactivation domain, you get a lot of papers.
Thank you.
Yeah, one question.
Hello, sir.
Yeah.
Sir, here.
On
This
the
side.
other side.
Yeah.
Thank you for the talk, sir. So I was wondering that as an academician, when you have a technology that you have launched, and you have, let's say, like you showed an example, your MOU expired, and then your patent got expired also. What is the scope of still profiting off the technology that you created?
I don't know. Like I said, I am an idiot. No, I hardly made 10 lakhs out of the hepatitis B vaccine technology. Of this 10 lakhs, 60% came to me, 40% went to the institute. Okay, only 60% comes to investigator. And about tech transfer fee also. See, at that time, this kind of academic industry was never happening. No, I was more enthusiastic to give the technology to a company so that it takes off. No, I am contributing to nation development and all that. No, but at that time, even there was no support system available in the institute. Today we have a called IPTEL, Intellectual Property Transfer Office or something like that. They draft MOUs for you. They negotiate with companies. For example, I said we are now negotiating with Biocon, for example, who are actually making insulin. We are now going to just draft the MOU to see, can we give a glutamate-inducible expression system to them, so that if it can they make insulin from both glutamate-inducible promoter and methanol-inducible promoter, so that using the same infrastructure they have now, can they double insulin production? Instead of making from one promoter, if you make from 2 promoters, will you double insulin production? We are now signing MOU. But I don't know the commercial potential. I don't know how much company is willing. I am a scientist. I am not a marketing guy. I don't know. These are done. I said it is up to the institute to do all this. I have no idea. But today, after doing all this, I am looking like an idiot. Because instead of saying the MOU is valid for 7 years, if I had said MOU is valid for 15 years, I would have got 10 crores instead of 10 lakhs. So nobody guided me at that time. So you have to be smart.
Okay, maybe
companies are smarter than you.
We'll take one last question over there.
Hi, hello sir. Excellent talk and really inspiring to see the actual research that goes into industry and creates really fruitful results. So I have a question about like going from here, where do you see the, in Indian system, where do you see the lack, whether in academia, like in basic research, or developing more and more industrial facilities, or that knowledge system, or that supply chain, where can the barrier be present, where India still is a little behind in order to...
we are a developing nation still, right?
Yeah.
Our economy is what? I mean, 10% of US economy, no? Compared to China, we are nowhere. They are all developed countries, no? They all made. But they all, China made money only by indigenous technologies, no? Apple went and made mobiles in China. It was a manufacturing economy to begin with. They did not have. But Japan, for example, was a manufacturing economy. They did not do any innovations. What innovations came out of China? Not much. They all made, because their labor is cheap, they can make things cheaper. They bought innovations from US, because today if you want to make profit, you have to compete globally, you have to make things cheaper. That's where India also has an advantage. That's why the government is pushing Make in India programs. Because if you economic development is very important. How can I pump money into innovative research if government has no money, right? Government has to make money first, no? And how many people are paying taxes? Hardly 5%, right? Everybody is cheating, right? So government, if government has to make money, they have to create jobs, they have to manufacture, they have to export. That's where we are now. But nothing prevents you from innovating, no? Without giving, these are like two eyes, no? I cannot say left eye is more powerful than right eye. Basic research, applied or translation, both has to be done. That's why I give an example. For example, I never thought understanding glutamate metabolism yeast can develop a new expression system. That's an example of an innovation, no, which came out of basic research. I wanted to understand how glutamate induces the synthesis of glutamate dehydrogenase and PepsiK. In the process, I developed an expression system. Basic research, when you do, you never know when you can get, but you can get breakthroughs only through innovative research. But both are important. Basic research, indigenous research, innovative research, all are very important. But some people are good in basic research, some people are good in doing both, some people are good in this. It's a mixed bag. You have to decide what you are good at and do that.
Thank you, sir.
Thank you so much for taking all those questions and for your insightful talk. I call upon Professor Jyoti to give a small memento for being here on our National Science Day and spending time to explain all his journey of making the vaccines, the goods and the bads, so that everyone knows. Thank you so much, Professor Rangarajan. Thank you. So now we will break for a break of a coffee for about 10 to 15 minutes, not more than that, and then we will resume the program thereafter. So I think we'll come back around 11:40. We'll be back in this. Please, everybody come back at 11:40. Thank you. So let's continue the National Science Day. I'll call on Dr. Meenal to please introduce our next speaker, Dr. Vinay. Please, Meenal, would you come on stage? It is my pleasure to introduce Dr. Vinay Kumar Rao for our faculty talk today. Vinay is one of our new faculty members here at MIRM, and he obtained his doctorate degree from National University of Singapore under the guidance of Professor Reshma Taneja, a stalwart in cancer biology and epigenetics. An assistant professor from JSS, he has multiple years of postdoctoral experience and at Singapore, and industrial experience from Stempeutics Research Private Limited, and his extensive contribution in his multiple publications in the field of epigenetics, specifically neuronal differentiation, cancer biology, and other works with muscle biology and mesenchymal stem cells. I want to welcome you, and it's a very happy moment to welcome one of new faculty members to listen to his talk. Welcome, Vinay.
Hello, can you hear me? Can you guys hear me? I hope I'm audible now. Yeah. All right. Thank you, Meenal. Thank you for the nice introduction. I would like to thank Professor Jyoti and Shweta for the opportunity. As you all know, I started my lab just 2.5 years back as an independent faculty. And my lab has been interested in understanding the basic biology of how cells differentiates, right? In particular, we are interested in muscle cells. How do these muscle cells differentiate to form multinucleated myofibers? In general, what we understand in our lab by doing experiments is to know what are the DNA modifications that happens, what are the changes that happens at the histone level for a cell to differentiate from an undifferentiated state towards a differentiated cell. On the other end of the spectrum, we are interested to identify the molecular targets that are deregulated in several muscle disorders. So one of the groups work on muscle atrophy, which is seen in cancer condition, which is called as cancer cachexia. Another student is working on muscle atrophy in diabetes. We also work on a rare cancer, which is called rhabdomyosarcoma. It's a cancer of skeletal muscle origin seen in children. And we are trying to see what are the molecular targets if we can identify and be able to treat these kind of skeletal muscle disorders. In today's talk, in the first half, I am going to talk about the epigenetic regulation of myogenic differentiation, which we have published. In the second half, I will be talking about our recent unpublished work, which is on muscle atrophy. You all know skeletal muscle is central to everything we do in life, be it running, dance, or whatever the activities, skeletal muscles are very crucial. And it's the largest tissue in the body. Approximately 40% of the body mass is covered by skeletal muscle. And as we age, the function of these muscles decline. It is one of the highly regenerative tissue in our body, and it is highly regenerative because of the presence of satellite cells, which are called as muscle stem cells. Actually, it was in 1961, Alexander Moreau, he was the one who first identified that there are resident muscle stem cells, which he termed as satellite cells because of its position in the basal lamina. So these cells are quiescent in nature. Upon injury, whenever we fall, we trip, when there is a muscle sprain, these cells, they get activated. They enter into the proliferation state from the quiescent state. Once they activated, they turn on muscle genes. They later on become fused to form multinucleated myotubes. So in the lab in vitro conditions, we culture these muscle cells as myoblasts. They express certain transcription factors which are important for their proliferation, such as E2F1, In addition to that, since these are myoblasts, they express MyoD, myogenic determination factor 1. MyoD is called as a master regulator of myogenic differentiation factor, myogenic differentiation, because if you take MyoD transcription factor, if you put it into non-muscle cells, it has the ability to convert those non-muscle cells into muscle cells. So it's a master regulator of the differentiation program. So under proliferation conditions, it expresses E2F1 and MyoD, and when we provide them the differentiation cues, these cells exit the cell cycle. So for a cell to differentiate, the first and foremost thing that needs to happen is it needs to exit the cell cycle, which is an irreversible cell cycle exit. Of course, there is also something called as reversible cell cycle exit, where the cells can enter into a quiescent state. But in today's talk, we'll be focusing more on irreversible cell cycle exit. So when we provide them the differentiation cues, these cells exit the cell cycle. They turn on certain transcription factors, certain genes such as P21, myogenin. They start aligning. These cells, which are undifferentiated cells, they start aligning. Once they align, they later on at some point fuse to form multinucleated myotubes, which are characterized by the terminal differentiation markers such as MRF4, myosin heavy chain, or troponin T. So the entire process of proliferation and to the differentiation is controlled by what we call as myogenic regulatory factors such as MyoD, P21, MRF4. So in addition to these myogenic regulatory factors, you have epigenetic mechanisms that are in place to control this proliferation and differentiation aspect of muscle cells. Now, although I told you MyoD is expressed in myoblast, it is not able to turn on the differentiation unless we provide them the differentiation cues. And this is because if you look at the promoters of muscle specific genes, and in fact Professor PNR nicely explained you how does the promoter gets activated, what are the transcription factors. So if you look at the promoters of muscle specific genes, you have MyoD binding onto the muscle promoter, but now it is in complex with the co-repressors such as G9A, HTAC, Su39H1. These are co-repressors which mediates H3K9 dimethylation marks, which are generally responded for repression of a gene. Whereas if you look at E2F1, E2F1 is in complex with a activator, co-activator such as PCAF or P300. That's where the genes are getting expressed and cells are able to proliferate. So when you switch them to differentiation medium, there is a switch in this co-activator co-repressor complex along with the transcription factors. So now MyoD is in complex with P300 and PCAF, thereby it mediates acetylation marks which are required for gene expression, while E2F1 seems to be in complex with a co-repressor where it is not able to turn on its gene. Our lab has been interested to understand role of G9A in the context of this muscle differentiation. G9A is a histone methyl transferase. Basically, it is known for mediating methylation marks on histones. And it functions as a repressor because it is mediating H3K9 dimethylation marks. It is very important for the development because loss of G9A leads to embryonic lethality. And this is the domain structure of G9A. It has an anchoring repeat domain where it helps in protein-protein interaction, whereas the set domain is the main business domain which is responsible for H3K9 dimethylation marks. Not only histones, G9A is known to mediate methylation on non-histone substrates such as certain transcription factors as well like MyoD. So now before we started this project, what was known in the field was G9A, when you overexpress G9A, which is a histone methyl transferase, it inhibits the myogenic differentiation program. Even from publications from our group as well as some other publications, they have shown that G9A when overexpressed inhibits myogenic differentiation program. Like it is in complex with MyoD, G9A methylates MyoD, methylation of MyoD functions as a repressor of its activity. What was not known is whether, since G9A is expressed in proliferating cells, how does it affects the proliferation of cells? Since I told you that cell cycle exit is important for myogenic differentiation, does it have a role in cell cycle exit? So these were the questions which were there when we started this project. So we started asking what are the mechanisms by which G9A regulate myoblast cell cycle. To understand that, first what we did was we took these myoblasts, we knocked down G9A using siRNA approach, we did a simple microarray analysis, and what we found was the most function that was altered was cell cycle development disorder and cancer. Genes related to these pathways were significantly altered. and we took a bunch of cell cycle genes. If you look at here, for instance, P21 and RB1, which are critical for cell cycle exit, were all upregulated. That means when I knock down gene NA, they got upregulated. That means gene NA inhibits their expression. Where at the same time, there was a bunch of genes which were all downregulated. So that means gene NA is required for their expression. So somehow gene NA is able to regulate genes involved in cell cycle. We asked next, does it affect the proliferation of cells? If it is affecting the genes involved in cell cycle, does it affect the proliferation of cells? For that, we did gain of function, loss of function studies to understand its role. So for that, we took primary myoblasts from mouse. We did a knockdown experiment, and we did a simple flow cytometry. This is the knockdown efficiency. We did a simple flow cytometry experiment, which tells that when you knock down gene NA, the number of cells which are present in S phase were lower. We also followed it up by doing a BRDU incorporation assay, BRDU chase assay. We found that when you knock down gene NA, actually the number of cells which are proliferating are lesser. We also performed overexpression system by retroviral infection, which I'm not showing you here. We found the opposite of what we saw when we knocked down, which indicated that gene NA knockdown reduces proliferation of myoblasts, and it is required for proliferation of cells. So now, as a part of validation from our microarrays, we took those myoblasts and again knocked down gene NA. We looked at the expression of P21, RB1, which are required for cell cycle exit. At the same time, we also looked at the expression of E2F1 target genes such as cyclin D, cyclin E, DHFR. They were all downregulated, whereas cell cycle exit genes were upregulated. We know that gene NA is a histone methyltransferase, so we blocked its activity using UNC compound to check whether it replicates what we saw in SIG9A. So when we blocked its activity, we found that P21 expression increased, RB1 expression increased. While it was surprising for us, none of the genes which were E2F1 target were regulated. Like here, when you knock down, they were downregulated, whereas when you inhibit its activity by the compound, their expression didn't seem to change. So somehow, they might be methyltransferase, gene NA might be functioning in an independent methyltransferase independent manner. So now you have a two scenario where gene NA could be functioning as a repressor of P21 and RB1, which could be a methyltransferase dependent, whereas gene NA could also function as an activator of gene expression. Remember, I told you that gene NA is a repressor, but here we have a scenario where it might be functioning as an activator. So we wanted to ask, does it really function as an activator? Because when you knock down, some of the cell cycle genes were downregulated, but when you inhibit its activity, it was not changed. So in order to do that, first we wanted to check, is it a direct target? So we want to perform some chromatin immunoprecipitation assays to check whether they directly bind to the promoters. So we performed chip analysis, looking at gene NA occupancy on these genes. For instance, P21 and RB1, gene NA occupied on their promoter, and correspondingly there was increased in methylation marks on these promoters, which indicates that it's a direct target, whereas acetylation did not change much because we knew that these were direct targets and gene NA does H-recombinant dimethylation. We looked at E2F1 target genes. Again, gene NA was bound onto these promoters. So that means they are direct targets. However, there was no H3K9 dimethylation marks seen on those promoters. Instead, we found that there was increase in acetylation on these promoters. So now gene NA being a histone methyltransferase, it was mediating acetylation. So the question is, how can it mediate the acetylation? Is it in an activator complex? So for that, we asked, does it interact with E2F1? If it interacts with E2F1, is it in complex with PCAF as well? So we asked this question and we wanted to test it out. So we performed a co-immunoprecipitation assay where co-IPs, we overexpress gene NA flag, flag PCAF, gene NA, and E2F1. In fact, they were in a complex. E2F1, PCAF, and gene NA were in a complex. And then we asked more like whether gene NA recruits PCAF to the E2F1. So we took gene NA overexpressing cells, which are PBAB gene NA cells, stable cells. And in the presence of overexpression of gene NA, you have higher association of E2F1 with PCAF1. And in fact, even if you look at their promoters, cyclin D1 and DHFR promoters, there is higher occupancy of PCAF when you overexpress gene NA. So right now what I have told you is gene NA is in complex with PCAF1 and E2F1 in myoblast. But at the same time, what was known is gene NA is interacting with MyoD. And both these are myoblast. In the same cell, you have two complex. One is gene NA is complex with E2F1 and functioning as an activator, whereas it is in complex with MyoD and functioning as a repressor. So now how can this happen, right? So we thought through this and we hypothesized that we know that E2F1 activity is higher at G1S boundary or in G1S phase. So we thought it could be possible that G9A interaction with these two transcription factor is cell cycle dependent. So for that what we did, we wanted to test out this hypothesis. So we took synchronized cells, we synchronized cells at G1S boundary and the other cells at G2M boundary by nocodazole and hydroxyurea. We took these two populations. Now we asked, where does G9A interact with MyoD and E2F1? So in fact, when we did these co-IP experiments, we found that G9A interacts with E2F1 more at G1S stage, whereas with MyoD at G2M. In fact, G9A interaction with MyoD, after we published this, it was known that repression of MyoD should happen at mitosis because it should not lead to the differentiation. So this was in line with what we found out, that the interaction of G9A is with these transcription factors are cell cycle dependent. So in summary, what I have told you is in proliferating cells, in proliferating skeletal muscle cells, G9A exists in two complexes. One, it complexes with E2F1 and PCAF, and it mediates acetylation marks through PCAF on proliferating genes. Thereby, it functions as a activator of gene expression in a methyltransferase independent manner, while it is also in complex with MyoD1 and HTAC, which is again cell cycle dependent, depending on at what stage the cells are. They inhibit the cell cycle exit, because if you remember, P21 and RB1 are the genes required for cell cycle exit. So they inhibit their expression, and thereby it acts as a master regulator of myogenic differentiation program. In one side, it is helping the cells more to proliferate. On the other side, it is inhibiting the cell cycle exit. So if both these things are happening, it's functioning as a master regulator of myogenic differentiation program. So this was published in NAR. So now I'm going to switch the gears to talk about more of our recent work, which is still unpublished. So where one of my students is trying to understand the mechanisms underlying hyperglycemia mediated muscle atrophy. So if you look at the muscle atrophy, basically it is loss of muscle mass, loss of muscle strength, which is seen not just in diabetes, but in many cases. For instance, even aging, as we age, our muscle strength, muscle mass decreases, which we generally refer to as sarcopenia, right? So not only during normal aging, you have in the context of cancer, diabetes, sepsis, denervation, genetic diseases. In several of these cases, muscle undergoes loss of its function as well as mass, right? So this is called muscle atrophy, which leads to decrease in muscle mass and strength. You have a locomotion disabilities because of that, because 40% of the body mass is involved in the locomotion, and you have reduced muscle regeneration as well. I told you that muscle has a very high capacity to regenerate, but as and when we age or during these insults, you have reduced regeneration. So muscle atrophy is characterized by reduction in muscle mass. As I told you, if you look at the histology, you have a very smaller myofiber size, and under normal conditions, under normal homeostasis, physiological conditions, you have a balance between muscle protein breakdown, the number of proteins that get breakdown, and the synthesis. So under normal physiological condition, you have this balance, homeostasis maintained, and that's where you have a healthy muscle. Whereas under atrophic conditions, there are several reasons, not just, I'm not saying it's just hyperglycemia, it could be inflammation, oxidative stress, increased expression of ubiquitin proteasome pathway genes, autophagy. Many of these things, they ultimately converge on to turning on FOXO group of transcription factors, or NFKB, which leads to increased muscle protein breakdown. So there is an imbalance in the muscle protein breakdown and muscle protein synthesis, which leads to atrophy.
So let me quickly highlight a few points for the participants to keep in mind. There will be a 7-minute timer displayed on the screen below, and another bell will be rung at a 9 minutes mark. Each talk will be followed by a Q&A session from the audience, and students' participation is encouraged. With that, let me call upon Ms. Supriya Bhatt to give a presentation.
So, good afternoon, everyone. Okay. So, I'm happy to present my work based on PVA chitosan hydrogel as a biocompatible scaffold for hepatocytes function and regeneration. So, jumping on to the introduction, as we all know that liver plays a major role in metabolism, detoxification, protein synthesis, and cholesterol synthesis, and it also has a self-regeneration capacity. But in few disorders such as hepatic cancer, liver cirrhosis, and fatty liver, the self-regeneration capacity decreases, and that leads to liver failure and death. So the gold standard treatment is liver transplantation, but due to donor scarcity, high cost, and post-operational complexities and immune rejections, this treatment poses a challenge. So to overcome this, researchers are finding a new approach, that is liver tissue engineering based on 3D bioprinting technologies. So I'll tell you why 3D culture is important. We know that in 3D cultures, it mimics a physiologically relevant environment as in vivo conditions and has better cell interactions. So in our studies, we are going to use 3D bioprinting technology to overcome this challenge. We have divided our methodology into two sections, that is preparation of hydrogels and checking the compatibility of the 3D printed constructs. So we have used, in our study, we have used PVA and chitosan to prepare our hydrogels. I'm going to tell you why we have used PVA and chitosan. PVA, as we all know, that it's a synthetic polymer and FDA approved, and it has high mechanical strength. And chitosan, it's a natural polymer derived from exoskeleton of crustaceans, but it has similar GAG structure to the ECM components of the liver. So we are blending these hydrogels and making a cross-linking using 3 different cross-linking methods, and characterization using SEM analysis to check the porosity of the scaffolds, and FTIR to check the cross-linking efficiency, and viscosity measurements to check the mechanical strength and viscosity of the scaffolds. And further, we are using a 3D bioprinting technology, that's extrusion-based 3D bioprinting, to fabricate these scaffolds into 3D constructs. And upon these 3D constructs, we are going to check the biocompatibility using primary rat hepatocytes, and we are going to check their metabolic functions and also cytocompatibility studies. So coming to our first part, we prepared hydrogel using chitosan and PVA and blended in equal ratios and cross-linked with 3 different methods, that is freeze-thaw method, janipin, and glyceraldehyde. Freeze-thaw, it's a natural type of cross-linkers which forms hydroxyl groups between the hydrogels, and janipin, it's a natural cross-linker that is derived from fruit Gardenia jasminoides, and it forms amide bond between the linkages of the hydrogels. And glyceraldehyde cross-linking, it's a chemical cross-linking. It forms imine bonds between the hydrogel groups. And this is our cross-linked hydrogels. So the cross-linked hydrogels were further characterized for physicochemical and mechanical properties. We did SEM analysis to check the porosity and pore size and hydrogel morphology. We could see that in freeze-thaw scaffold and in janipin cross-linked scaffolds, the pore size was better, and there was an interconnected pores, which helps in cell infiltration, cell attachment, and nutrient exchange. And to check the confirmation of the cross-linking, we did FTIR analysis. We could see that there was a formation of hydroxyl groups in freeze-thaw method, and there was a formation of amide bonds in janipin method, and there was a formation of imide linkages in glyceraldehyde. This confirms our cross-linking. And to check and we did swelling ratio analysis and degradation. Swelling and degradation to check the water retention capacity and how fast the gels are going to degrade, or how long it can sustain the structural integrity. So in our swelling ratio and degradation, we could see that uncross-linked gels took up the water fast, and also it degraded faster compared to our cross-linked gels. So further, we moved on with the rheological analysis to check the mechanical strength and viscosity measurements. Upon cross-linking, the viscosity also increased and the mechanical strength was also increased, which is a key factor for 3D printing applications. So after checking all these parameters, we moved for 3D bioprinting of the scaffolds using extrusion-based bioprinting. And these were our optimized scaffolds of different cross-linking methods at 25 gauge needle and different pressures. So after optimizing these scaffolds, we wanted to check the biocompatibility of the scaffolds for hepatocytes regeneration. So we isolated rat hepatocytes using perfusion method, and after isolation, we did the confirmation using albumin and cytokeratin assay to check the albumin synthesis and epithelial morphology of the of the hepatocytes. After confirmation, we moved on with the cytotoxicity assays using MTT and live dead assay. In live dead assay, we could see that in 2D cultures, that's on TCPS, after day 5, there was a fibroblast morphology observed. That means the hepatocytes was differentiated into fibroblast morphology in 2D cultures. But on our 3D printed constructs, there was no such morphology differentiation, there was no such morphology change observed. And also in MTT assay, we could see that in freeze-thaw and genipin scaffolds, the cell morphology and cell proliferation was better compared to glutaraldehyde scaffolds. As we all know that glutaraldehyde is causes cytotoxicity to the scaffold. Similar results was also observed in live dead assay. And after all this analysis, we wanted to check the functionality assessments of albumin and urea, that is albumin for synthesis and urea for ammonia clearance of the hepatocytes on these scaffolds. So we could see that in albumin assay and urea assay, we could see that freeze-thaw and genipin had better functionality compared to glutaraldehyde scaffolds. So by all this, we could conclude that freeze-thaw and genipin had better cytocompatibility, and also in the mechanical strength and also in the physicochemical properties that showed better results. Scaffolds. So after all this confirmation, we prepared a hydrogel using the combination of PV and chitosan, adding DLM gel. Using the combination of PV and chitosan, adding DLM gel. Last was, we did, for these scaffolds also we conducted live dead and metabolic functions. In live dead, we could see that the gels mixed with DLM had better cytocompatibility, and also in the functionality assessments, we could see that albumin and urea synthesis was better in the DLM hydrogels. So as I conclude that our hydrogels was better cytocompatible, and I would like to acknowledge my guide, Dr. Manasanune, for giving me this opportunity. And these are the few conferences I presented, and these are list of my publications. Thank you.
We'll take questions quickly.
Yeah, just one or two questions. Anybody?
One or two questions. We have time. We have 2 minutes. Yeah. Okay. Any questions? All right. We'll move on.
Thank you.
Okay. Thank you. Restart. Laser pointer. So we'll now call upon Mr. Govind for his talk.
So the whole idea of extracellular ATP in the cell extracellular space, like how that is important in a normal physiology or in a disease physiology, is very important. Like even if recent studies show that the circadian rhythm also maintained by the pumping of extracellular ATP to the cell outside. And that has a very important role in the normal physiology as well. When there's a stress or inflammation, the cells will secrete an immense amount of extracellular ATP. And ultimately, in the case of a cell, for a death or a survival determination, it is important that the ATP has to be cleaved. And the cleavage will happen in such a way that it will finally converted to adenosine by a series of enzymes. And CD73 or the ND5 is one of the unique enzymes that convert the AMP to adenosine. And it is expressed in most of the cells. And coming to the structure of the CD73, and it's a very highly conserved surface enzyme which is conserved across the species. And as you can see here, in the B figure, where the bacterial ND5 is completely superimposing with the human ND5 monomer, that indicates it's very conserved. And how the AMP will interact is like there is 2 phenylalanine ring which will actually act as a stack, and where the AMB is stocked in between these two phenylalanine ring, and one asparagine 390 residue is there, which actually have a hydrogen bonding. And it is very interesting that these two residues are highly conserved across the species, is highly conserved or semi-conserved across the species. So what happens when the CD73 is not there in the humans? So just imagine a person is living from his childhood to youth, thinking that he's absolutely normal, and suddenly in the early adulthood, he or she start experiencing joint pain or joint calcification, and thinking, okay, it's kind of an osteoarthritis because it will actually mimic the osteoarthritis symptom. And later, he or she develop lower extremity calcification. So if it's very fortunate, then they will understand that this is because of the mutation in CD73 through a genetic analysis, where it is marked with an homozygous recessive disease called arterial calcification deficiency due to CD73 deficiency. But unfortunately, this particular phenotype is not there in the mouse system. Mouse system doesn't exhibit this vascular calcification. And the treatment, when the first case got reported in 2012, and following these years, the clinical trial showed that etranol treatment is not successful in treating the ACDC. So even though this is a rare disease, there is like more occurrence among us, maybe considering it as an osteoarthritis. And we don't know where the exact calcification is happening, whether it's the endothelial cells getting calcified or the smooth muscle cells getting calcified. That mechanism is not yet known. So based on this, under the guidance of Jyoti ma'am, we started this work to understand the mechanisms related to ACDC disease. So what we started is, we started with knocking out the ND5B in human aortic endothelial cells, where we, using CRISPR-Cas9, we did a complete knockout of the ND5B, and we put it for the RNA-seq analysis, where we are seeing a lot of cell adhesion molecules or angiogenesis molecules are downregulating, and we further put it to test in a surrogate angiogenesis assay. As you can see here, in the mutant, there's an impaired angiogenesis. And another thing what we proposed was like whether it can be a trans differentiation, or it might be an endothelial dysfunction, or it could be a endothelial to mesenchymal transition. So what we saw is like we saw the Run-X2 induction, spontaneous induction in the endothelial cells post-knockout. So this is the one part of the study like which we are pursuing. Apart from that, like I told, like this is highly evolutionary conserved. So, and there is no one yet tried to mutate those two amino acids which actually act as a clamp for the AMB. So what we thought is like thinking it as an it's an opportunity. What we tried is we tried mutating these two amino acids and see whether only these two amino acids are required to curb the activity. So this way we will be able to tell whether what is the enzymatic activity dependent role, or the activity enzymatic inactive roles. So what we did is we had the overexpression construct. We first mutated one of the phenylalanine residue to valine, where we still saw the activity on the surface. Again, the other way around also we just mutated the 417 residue alone, phenylalanine to valine, where there also we saw the activity. When we mutated both the phenylalanine residue that stacks the AMP to valine residue, we saw complete reduction in the activity. As you can see here in the activity assay, with both the mutation, the activity is curbed. We further checked, so we want to know whether there is an activity dependent and activity independent role for the CD73, whether it can interact with any other molecules on the extracellular matrix. That might be a reason for the angiogenesis defects what we are seeing. So in order to this, we put it to test to understand whether this is coming to the surface, this protein is coming to the surface or not in the HEK system, where the cells don't express CD73 as such. So what we are seeing is, this is our overexpression system, where we are seeing a surface expression of CD73, where in the dual mutation also we are seeing a surface expression of CD73, but there is no activity. And we further validated it with the flow cytometry analysis. And this is a novel attempt which can be extrapolated to our knockout cells. And understand how this is affecting the effect. And when we started this study, there was 12 patients across the globe, and in 2021, the first Indian patient got reported. And we were lucky enough to trace back him, and we had a repository of his samples in the lab now. And we are trying to get an organotypic model out of the system, try all the possibilities, because there are multiple forms of CD73, having exosome-bound form, the cleaved form, and there's recently reported a circular form and an inactive form. So understanding what exactly is doing what, so that will be a crucial thing to pursue. And of course, this is a work in progress. And I would like to extend my thanks to Jyoti ma'am and my lab members, Anju sir and his lab members for the inputs. And of course, supporting financial support by DST Inspire, ICMR, and MAHE. Thank you.
We'll get questions now.
It's just a simple question. I'm surprised this occurrence is very low. I mean, and lately identified in the sense that I wonder why.
Sorry, ma'am?
The occurrence is very low. So was it there earlier itself or just happened to identify it recently?
So, ma'am, the case first came in 2012. So, ma'am, it's because ultimately this thing, this disease need a genetic analysis, and you know the expense of the genetic analysis in India is very high. And if the patient, so what happens is when we see the patient, so he will show the classic symptoms of osteoarthritis, having the inflammation, heat generation in the joints and everything. So it is, most of the time, I think it might be misunderstood as osteoarthritis.
Right.
And interesting part is like even osteoarthritis exudates also show lower levels of CD73.
Right.
That study is there. So it's obvious, it's CD73 is mostly studied in cancer, and the normal physiology we have to understand like what exactly is happening.
But are there other bone deformities that possibly have this mutation? Is there any?
In mouse system, they have shown the bone deformities, and unfortunately, this particular disease phenotype, the calcification events, is not there in the mouse system. That makes it a huge opportunity to understand, and of course, this is a rare disease, and we have to repurpose the drugs.
Right.
So for that, a system is needed, and the system is not in place. So our aim to develop a system that where we can test the things out.
Okay, good.
Can you rescue this phenotype by just adding AMP, that means?
So that's where the other forms also comes into picture. Like one intracellular form is there, which only 2 articles are till the date where they have shown it, the intracellular form will bind to this active form and take it for proteasomal degradation. And even the active form also cleaves out as an active protein, and it can go to the circulation, and it can be bound to exosome also. So if we delineate each form and understand what exactly it is doing, then we will be able to tell whether it's an activity-dependent effect or the activity-independent effect.
Okay.
And there is one circular form also recently got published. So that also regulates the surface form of the CD73. So it's like 3 forms. Okay. It's like a triangle. Sir, as such, there is an extracellular ATP reservoir is there. When there's an inflammation of like any stress or anything, more ATP can be released, and it will gradually lead to cell apoptosis. Yes, but the patient, we don't know, because it's an early adulthood disease. It might be because of the shear stress that is happening. So we need to have a 3D model to understand the exact thing, because it's a blood vessel thing. So, and the endothelial lining is breaching or not, we don't know. Whether the smooth muscles is going to a contractile to synthetic phenotype, we don't know yet.
Thank you so much for the talk. Now I would like to invite Mr. Krishnamurthy for the next presentation.
Good afternoon to all. Myself Krishnamurthy, and my today's presentation entitled Aberrant EEG Microstates in Parkinson Disease Patients under the guidance of Dr. Anand. A brief introduction about EEG. So EEG is an acquisition of electrical activity of the neuron in the brain by placing the electrodes over the scalp. So now these recorded oscillations, so that can be defined as a non-overlapping states. So each state is characterized by unique topographies. So here topographies, how electric potential is distributed over the scalp, so that can be represented by two-dimensional graph. So that is called as topographies. So now these topographies, it's keep changing throughout the EEG recording, but still, so that is stable for 80 to 120 milliseconds. So those stable topographies is called as EEG microstates. So EEG microstates conventionally classified into 4 types. Microstate A, auditory network, B, visual, and C, attributed to default mode network, and D for attention network. So when subjects are at rest, so they are not doing any task, so then default mode networks will be the active one. So posterior cingulate cortex, anterior cingulate cortex, insular cortex are the main hub for default mode networks. And attention, when subjects are involved in any cognitive oriented task, so that time that attention network will be the dominant one. Intraparietal sulcus, prefrontal cortex are the main region for attention network. So microstate C is called as task negative network, and microstate D, task positive network. So EEG microstates represents, it's a short lasting periods of synchronized activity of large scale brain network, and any disruption in these networks, so that leads to neurological or neuropsychiatric disorder. So EEG microstate analysis would provide basic understanding of progressive deterioration of underlying neural network over the progression of PD, and also provides neurobiological correlations for neuropsychiatric as well as cognitive comorbidities. So objective of current study is to investigate the alteration of EEG microstates in Parkinson's disease patients when compared to normal controls. So we have used EEG data of 28 normal control. So microstate C for default mode network, and microstate D for dorsal attention network. So this graph shows parameters of D is positively correlated with the disease duration. So as the disease duration, ED patients. So I acknowledge Manipal Academy of Higher Education for their financial support. Thank you.
Do we have any questions? Can I ask a question? Okay, so what is the time span for these microstate that you measure in the EEGs?
So that is 80 to 120 milliseconds.
And how is it that you're able to, the kind of data you extrapolated from that much duration, how were you able to do that? Like,
no, that
what is the
separate tool is there to access that data. So we are not explicitly doing anything. That is separate tools for to extract EEG or to categorize into different microstates. So with human eyes, we cannot detect the milliseconds durations. So that is seconds only we can detect as per human. But this can be done through separate software.
Krishna, I had a question. Can this be considered as diagnosis in the sense of early stages of Parkinson's? Can be used as an identification mark?
Yes, madam. But in this database, we have subjects from 1 year to 10 years is there. So we have only 28 subjects. So that can be applied to early detection or something like that.
Okay.
Yes. This one.
What are the parameters you are getting transition from
yes,
A to D? Is it very specific for Parkinson's, or is schizophrenia patient if you take
No, it's actually in microstates other papers like PD papers, they have shown reverse results like increased microstate C and microstate D. But in this particular database, it is they did not.
No, what I mean is, you take the dopaminergic neuron degeneration leads to multiple things. Along with PD, you have schizophrenia, you have ADHD, so all these things are there. So is it specific only to PD, or even in schizophrenia you see the same parameter, I mean same?
Yes, that is possibility is there. They have explained in their own way to justify their results. But for that purpose only, we have used the alpha power as a justification to make it more clear. So hypervigilancy, that is a common phenomenon in PD as well as some other diseases also. So that is how we try to prove that one.
Other psychiatric conditions can be considered monitored by this method?
I have not come across those papers, but schizophrenia, most of the papers are there.
Okay.
So in that it's there, I think. There specifically, I am not sure, I can't recall that one.
Okay. Other psychiatric diseases?
Okay. Yes. Thank you all.
Thank you for the presentation. I would like to call upon Ms. Abhinaya Raghavan for the next talk.
Hello all. So today I'll be presenting on astroglia proliferate upon biogenesis of tunneling nanotubes via alpha-synuclein dependent transient nuclear translocation of focal adhesion kinase. So what are these tunneling nanotubes? Tunneling nanotubes were discovered in 2004. They are a recent discovery, but ever since then, in the last 2 decades, there are several reports of their implications, especially in neurodegenerative diseases. So this paper here, science paper, shows the first time where they were discovered. They are thin membrane structures between 2 cells. So, so there are several ways through which prion proteins spread in the system. So the common one is through endocytosis and through exosomes, but the recently discovered one is via the nanotubes. While we are interested in alpha-synuclein, why are we specifically interested in astroglia instead of neurons? It is known that neurons commonly release the aggregates which are later taken up by astroglia, and to know how this process happens and what is the importance of it is of therapeutic importance. So cell-to-cell spreading of prion proteins. So these are some of the reports that has shown the red labeled prion proteins like alpha-synuclein spreading from one cell to another. And these videos here, okay, it's not playing, show the transfer of alpha-synuclein protein. Okay. So there is a significant difference between the effect of alpha-synuclein in neurons and microglia. So in neurons, though it causes neurodegeneration, in glial cells, both in microglia as well as astrocytes, we see rescue of the cells. So how does this happen? So since there are no markers to identify tunneling nanotubes, one distinct characteristic is that it hovers between 2 cells. Unlike other cell protrusions, you don't see them on the surface. So when you do confocal Z-stacks, you can see the left panel shows TNTs which are hovering, and the right panel shows tumor microtubes which are attached to the surface. So one significant observation we noticed is upon alpha-synuclein treatment in early time points like 3 and 6 hours, we see the tunneling nanotubes formed in huge significant numbers, which then reduces later. And these are some videos where we have seen both mitotracker and lysosome encapsulated lysotracker moving, alpha-synuclein encapsulated lysotracker moving through the cells. The videos are not playing, I'm sorry. And so, yeah, sure. So because we saw movement of organelles, we are interested to know what is exactly happening to the organelles. So when we stain for JC1, which shows any changes in the mitochondrial membrane potential, we see significant mitochondrial toxicity in 3 and 6 hours, which is later recovered in 24 hours. Not just that, we also see reduction in the lysosome accumulated with alpha-synuclein in 24 hours. So how does the cell recovery happen? So because we were doubtful that if it is just movement or actual transfer between two cells, we transfected two different populations, one with red mitochondria, another with actin, and we can see that the cells, the mitochondria from the green cells are present in, mitochondria from the red cells are present inside the green cells. So this confirmed that there is transfer of mitochondria from one cell to another through the tunneling nanotubes. And because we saw mitochondrial transfer, we also checked at ROS levels, and we saw that there is an increase in the ROS levels, which is also later significantly reduced. And we also did a control experiment without actual physical contact of the cells, where we did not see much transfer. So then, because of the involvement of ROS, we looked at cellular senescence. So while we did not see any changes in the irreversible P16 senescent marker, we saw a significant change in the P21 marker. We also saw other senescence markers go up in 3 hours, just like fragmented nuclei and beta-galactosidase. Another very important observation we had was rounded cells. So therefore, we checked at the focal adhesion kinase, and interestingly, in the cells which are connected by the tunneling nanotubes, we saw translocation of focal adhesion kinase to the nucleus. So because we know that this could actually remodulate the ROC pathway, we wanted to look at ROC pathway because TNTs are primarily actin structures. So there is a remodulation of ROC. You can see that in 3 hours, the ROC2 levels go down, which are then upregulated. So we believe that this is the pathway through which these TNTs are formed, biogenesis of TNTs. So apart from the cell survival, what was most interesting is because we know that ROC inhibition can activate ERK pathway and lead to increased cell proliferation, we looked at cell proliferation. And you can see that both with Ki67 marker and MTT assays, we saw a significant increase in cell proliferation at 24 hours. So the cells were not just surviving, but actually entering a hyperproliferative state. And we confirmed this through Westerns, where we can see an increase in phospho-ERK as well as NF-kappa beta. So we would like to conclude that the alpha-synuclein causes increase in ROS, which leads to toxicity in the cells, which are recovered through FAK translocation, which leads to ROC inhibition, which causes remodulation of the actin pathway, through which there is biogenesis of TNTs, which are helping in getting rid of the toxic burden in the cells. And the end of it, there is an upregulation of phospho-ERK and NF-kappa beta pathway, which is leading to increased cell proliferation. So I would like to thank my PI for this wonderful opportunity and all our collaborators without whom we couldn't have done this work, Dr. Ravi Manchitya, Dr. Shiv Raman, and Dr. Ravi Mudashedi. And my DAC members, Dr. Ramre and Dr. Shiv Raman, and Dean, PhD coordinator, my lab mates, and MAHE and ICMR for their fellowship. Thank you all. If you're interested more in our story, please check out our full paper, and I'm open for questions. Yeah. So, yeah.
So a quick question. It's really nice. So if you increase the ROS level without any synuclein treatment, then also you see the same thing? I'm here.
Oh, yeah, we haven't checked that yet, but we will look into that. Thank you.
Any other stressor you have looked into, or?
In this study, no, we have not looked into.
Thank you.
Abhinay, just right here.
Yes.
Is there a way for you to block these TNTs, the formation of them?
So when we used the cytokalasin D, which is an actin polymerization inhibitor, we saw a decrease in the amount of TNTs as well as decrease in the transfer of organelles.
Okay, so if you treat with alpha-synuclein and block them, then do you stop this proliferation that you see?
We haven't looked at proliferation, but in the cell numbers, there was definitely decrease in MTT, but we haven't checked Ki67.
For the cell proliferation, if it's really downstream, if the toxicity.
Cell numbers are less. cell viability reduces.
Viability rests, but I wondered about the phospho-ERK and all the other things that you showed
with
molecularly.
cytochalasin D, we have seen
Phospho-ERK levels are not getting
not
up.
phospho-ERK levels, but cell viability goes down.
Okay.
We have checked in MTT.
Okay.
Yeah.
Okay. Thanks.
Yeah, just a answer to Urmi's question. Actually, she not did, another student did. Certain amount of H2O2 increase for certain level, and Ross increased TNTs. There are multiple publications. You can see that. That is known. Oxidative stress and Ross.
Yeah.
So when you say that there is cell toxicity, you say there is like accumulation of these mitochondria, right?
Yeah. There is accumulation of mitochondria, there is increased lysosome size,
Yeah.
and there is a difference in the morphology of the cells.
Yeah. So now, when you're saying there is rescue because of the TNT, and they are being transferred to healthy cell, right? So what is happening to this healthy cell? Are they becoming pathogenic, or are they doing something to the engulfed mitochondria?
Probably the healthy cells are also helping with the defected cells. So the healthy cells, we believe, are taking up the load and helping in faster degradation of the defective mitochondria. That way, both the affected cells as well as the healthy cells are rescuing.
Do we have any more questions? Okay, I guess.
Yeah. Thank you. Yeah.
Thank you. Thank you for the talk, Abhinaya. Okay, with that, we have Ms. Sritama Banerjee with the next presentation.
Hello. Good afternoon, everyone. Today I'll be giving a talk on the topic of construction and destruction of PITX3, the transcription factor which is needed for the dopaminergic neuron differentiation. So, giving a brief introduction about dopamine and the roles of dopaminergic neurons. It is involved in reward, motor functions, pleasure, focus, attention, and motivation. And if there is any malfunctioning of the dopamine or dopaminergic neurons, it gives rises to some diseases such as ADHD, Parkinson's disease, and schizophrenia. So this makes it important to understand the development of dopaminergic neurons. So from the blastocyst, the 3 derm layers are formed, and it is from the ectoderm that the midbrain floor plate arises. And in the midbrain floor plate, there are multiple signaling pathways which are taking place that gives rise to the state-specific expression of different transcription factors, finally resulting in the generation of mature dopaminergic neurons, which is marked by the expression of tyrosine hydroxylase, or TH. Now, this TH serves as the rate-limiting enzyme for the synthesis pathway of dopamine. As I was talking about the different signaling pathways which drives the generation of dopaminergic neurons, literature shows gives a clear understanding about different pathways such as the SHH-FOXA signaling, TGF-beta, BMP, Wnt, and the exact mechanism of how they are actually participating in the generation of the dopaminergic neurons. Whereas talking about one important signaling pathway such as the FGF signaling, which is known to play a role in the generation of dopaminergic neurons, till date, we don't have a clear understanding of how exactly it is driving the generation of dopaminergic neurons. So to understand that, we have initially developed a model system to carry out our study. So from mouse embryo, we have dissected the midbrain, and we have characterized that we have taken the correct portion, which was subjected to the neurosphere formation, and that was plated on gelatin-coated plates to give rise to the progenitors. And under differentiation cues, we have generated the mature dopaminergic neurons that has been successfully characterized by the expression of SOX1 and nestin as the progenitor markers, and TUJ1 and TH, which serves as the mature neuronal and dopaminergic markers. So initially, we wanted to understand what is the effect of basic FGF, which is an important member of the FGF family, on TH expression. So as we can see here, both at the protein and the transcript level, we can see that upon basic FGF treatment, there was enhancement in TH expression, which was observed. So further, we wanted to understand what was the mechanism involved in basic FGF-driven TH upregulation, for which we had screened for different MB-specific markers. And we found that it was only specifically ZIC3 which was being upregulated. So ZIC3 has been previously shown to play a role in general neurogenesis and neural tube closure. However, what exact role ZIC3 had to play in dopaminergic neuron development was still unknown. So we initially wanted to see what is the effect of basic FGF on ZIC3 expression at protein level. So upon treatment with increasing concentration of basic FGF, there was a gradual increase of ZIC3 expression which was observed. Now, to understand the mechanism of ZIC3 in TH upregulation driven by basic FGF, we had done the gain of function and loss of function studies, where upon ZIC3 knockdown, we found that there was decrease in the expression of TH, and when we overexpress ZIC3 in the absence of basic FGF, it led to the upregulation of TH. Further, to further confirm whether ZIC3 is playing a role in derivation of TH expression, we knocked down ZIC3 we knocked down ZIC3 in primary neurons and overexpressed ZIC3 in the shRNA background, where we found that upon ZIC3 shRNA, there was reduction in TH expression, which was rescued upon ZIC3 overexpression. Now, this observation was further confirmed in vivo, where we found that in ZIC3 knockout mice, there was reduction in expression of TH. And when ZIC3 shRNA was stereotypically injected in the mice, we found that there was reduction in TH expression. So this shows that basic FGF via ZIC3 is regulating TH expression. So basic FGF acts on its downstream targets by multiple signaling pathways, that is the PLC gamma, the PI3K pathway, and the MAP kinase pathway. So next we were interested to understand what is the signaling pathway involved in basic FGF regulating the expression of ZIC3, for which we had individually inhibited these signaling pathways in the cells, and we found that it was only upon PI3K signaling that ZIC3 expression was being hindered. So upon PI3K inhibition, when we overexpress ZIC3, we found that upon PI3K inhibition, TH was going down, and it was being rescued upon ZIC3 overexpression. So this showed that basic FGF via PI3K signaling was driving the expression of ZIC3, which was further helping in the upregulation of TH. Now, further, we wanted to understand what was the mechanism involved in ZIC3 driving TH, whether it is directly binding to the TH promoter, for which we had screened for the presence of ZIC3 consensus binding site on the promoter of different MB-specific markers. And we found that it is not TH, but it is in the PITX3 promoter that there is presence of ZIC3 consensus binding site. So is it via PITX3 that ZIC3 is regulating the expression of TH? For which we had done the chip analysis, where we found that yes, ZIC3 does bind to PITX3 promoter, and when we did ZIC3 shRNA, we found both at the in vitro and in vivo level that PITX3 expression was going down. So further, to confirm our observation, we had done ZIC3 shRNA and PITX3 overexpression, where upon ZIC3 knockdown TH expression was reduced, and upon PITX3 overexpression, it was coming up. So further, to understand the effect of basic FGF on PITX3, we had treated the cells with basic FGF and found that basic FGF could successfully drive the expression of PITX3, and in the absence of basic FGF and also upon PI3K inhibition, we found that PITX3 level was going down, which was getting restored upon ZIC3 overexpression. So this completes the linear axis of basic FGF driving TH, where basic FGF via PI3K signaling was driving ZIC3, PITX3, and TH expression. So after understanding how PITX3 was actually being constructed, we were then interested to understand how PITX3 was being destroyed. So any protein undergoes so first, it was important to understand whether PITX3 was a stable protein or it was undergoing degradation, so which was confirmed by the cyclohexamide chase assay, where PITX3 showed to decrease in a time-dependent manner of CHX treatment. So further, it was important to understand whether it was by the autophagy or the proteasome pathway that PITX3 was being degraded. So upon chloroquine treatment, which is an autophagy inhibitor, we did not find any rescue in PITX3 expression, whereas it was only upon MG132 treatment, which is a proteasome proteasomal inhibitor, that we found that PIT-X3 expression was being rescued. Also upon proteasomal activation, we found that PIT-X3 was going down. So talking about UPS, there are three main enzymes which is playing a role, out of which our interest was to find out which is the E3 ligase which helps in the degradation of PIT-X3. So in silico analysis showed that TRIM11 to have the maximum hits. So further molecular docking studies also indicated that TRIM11 is probably the E3 ligase which is helping in the degradation of PIT-X3. So we knocked down TRIM11 and we found that PIT-X3 expression was being stabilized upon TRIM11 knockdown. So to conclude my study, basic FGF via PI3K signaling was driving the expression of ZIC3, which was binding to the PIT-X3 promoter, driving the expression of PIT-X3, which was further binding to the TH promoter and driving TH expression. So further, PIT-X3 was being destroyed via, with the help of TRIM11 by the ubiquitin proteasome system. So PIT-X3 is the key mediator of basic FGF signaling and driving the TH expression in MDA neuron development, and it is degraded by proteasome. So I would like to thank my guide, Dr. Anujit Kumar, Dean of MIRM, Dr. Jyoti Prasanna, my DAC members, my lab members, Dr. JP's lab members, and the funding agencies. Thank you so much. I would like to take up any questions.
PI3 kinase, are there any other inducers which can induce PI3 kinase, and same you can see?
Other inducers might be there, yes, but in our study we are mainly focusing on basic FGF as a representative of FGF signaling.
I just want to ask, in diseased condition, which part do you think is disrupted in the sense of the proteasomal degradation state? Is it the proteasome dependent, or it's part of the ZIC3, or is it basic FGF on the other side?
I think it's hard to point out at only one factor because it's a combination of multiple factors which can give rise to the diseased condition. But talking about PD in specific, if I talk about it, is the neurodegeneration which is playing a role. So for that, we went to see what is the development of the DA neurons to better understand the neurodegeneration part.
Okay.
Do we have any more questions? All right. With that, thank you so much, Shritama.
Thank you.
Okay. So we are at the last final stage of our flash talks, and next I would like to call upon Dr. Sandeep Mata for the next presentation.
Good afternoon, everyone. Good afternoon, everyone. So, every one of us know that healthy body begins with a healthy mouth. So here I would like to say one fact that our mouth talks to our body and our body talks to our mouth. So here if you see, the bacteria or any poisonous toxins which enter general circulation through the diseased gum and infect the rest of the body is a main key thing which I have taken here. So most of the people know that maintenance of oral hygiene is very most important thing. So any infected gums, if you see how the disease progression of the infected gums will lead to and interlinked with many of our systemic diseases, so such as like heart and lung disease, Alzheimer's disease, stroke, diabetes, colon cancer, obesity, premature birth. So many studies have proven that there is a clear-cut link between the periodontal disease, that is gum disease, with the systemic disease. With this, I have put my study to plan a regeneration effect for the periodontal disease. So in this context, my topic was regenerative potential of gingival mesenchymal stem cells and its application in periodontitis model. Myself, Sandeep, research scholar under the guidance of Dr. Anand. So here, I would like to brief about the periodontitis. Periodontitis is nothing but a chronic inflammatory disease which is surrounding the tooth. If you see the clinical conditions, healthy mouth, gingivitis, and periodontitis, there is a progression of disease stage-wise. So healthy to gingivitis, gingivitis to periodontitis. If you see the tooth structure, so teeth is protected by a protective barriers known as gingiva. Is a protective barrier in the oral cavity. So it surrounds the tooth where you have the bone and supportive structures known as periodontal ligaments. So integration of the gingiva to the tooth is very, very important. So in this context, balance of bone formation, resorption in healthy mouth, how it is maintained. To maintain that, so we should take our preventive measures. So in current interventions to treat this disease, we have non-surgical and surgical treatments. So though we have so many treatments till date, so regeneration of the lost bone, still there is a question mark. And we are using synthetic compounds, synthetic tissues and bone graft materials, and the output of it is not sure, guaranteed. So with this, we hypothesize our study that so use of autologous gingival mesenchymal stem cells can help in regeneration of our lost bones and also fight with the infection there. So here, so my choice of stem cells was tissue specific. So here I have compared a control one like dental pulp stem cells and along with the gingival mesenchymal stem cells. If you see the morphology of DPSCs and GMSCs were similar. There is no much change in the morphology of both DPSC and gingival MSCs. And also here we assess that the scratch wound assay between the DPSC and ST. So there also with the time period we didn't see much difference with the DPSC and gingival MSC. But here our main key contest is due to the periodontal infection. If you see there is a loss of surrounding tooth structure where bone, the oral bone is very important structure where we are seeing the bone loss. So here our key thing is to regenerate the lost bone. So in this context, if you see the osteogenesis, the process, so from mesenchymal stem cells to pre-osteoblasts, immature and osteoblasts and osteocytes. In this we have taken few markers to check whether our mesenchymal stem cells, tissue-specific mesenchymal stem cells, gingival MSC or DPSC will push towards the osteogenesis. So in this context, we have done osteogenesis differentiation for both DPSC and gingival MSC, where we found a small edge of more osteogenic potential in gingival MSCs. But not only this, if you see between dental pulp and gingiva, so to extract the dental pulp, we need to remove one tooth, first thing. So that is also slight painful procedure and you will lose with one of the tooth. But if in this context, if you see gingiva is robustly available inside your mouth. If you cut also, it heals without scarring. This is the best thing what we have. Throughout the age, healing is there inside the gum without any scarring. So when we analyze the Algerine red quantification between DPSC and gingiva, where we see significant increase between DPSC and gingival MSC. Likewise, ILP activity, one of the key marker for osteogenesis. So that assay also we have done where we have seen gingiva is more osteogenically little potential than DPSC. So I went ahead to use gingival MSC and we tried creating an animal model here, that is periodontitis model. So to create periodontitis model, so I have used feed material, that is rat feed material and nylon thread, where we have briefly anesthetized the visitor rat and we have placed at the below the gum. So if you see, I have taken one animal only, but one side I have untreated, other side is treated. So post 2 to 3 weeks, we could see with the sections also, there is a bone loss clearly evident here. With this, we concluded the preparation like formation of a bone loss model. And we went ahead to isolate, extract the gingival MSCs, and then these stem cells we have transplanted in the bone loss model. So in the bone loss model, what we have done is, we have created disease model on both the sides, right side and left side. So one side we have left as a disease control, other side we have treated with the stem cells. Post formation of bone loss model, that is 2 to 3 weeks, as we injected only in the left side. And we waited for 6 weeks, and we have sacrificed the animal, and we have taken a CBCT analysis to see whether bone formation with the stem cell injection of gingival MSC has happened or not. And we could clearly visualize here the bone formation is there, and the disease progression at the site of periodontitis model is stopped. Later, the cross sections we have done with HNE staining, and we can see this periodontal disease model, clearly this disruption of the fibers and the loss of bony structure and the stem cell injected sections compared to control. Same wise, to see the periodontal fibers, we have used one more staining, that is Mason-Rankine staining, whether how intact the fibers are there and how intact with the supporting tooth structure, that we can clearly observe in the cross sections. With this, the study outcome would pave way for translational approach in using appropriate oral derived autologous gingival mesenchymal stem cells to treat oral defects in clinical scenarios like periodontitis, traumatic jaw fractures. So exploring the possibilities of stem cell based cell-free therapy for oral diseases like condition media and exosomes derived from DPSs and gingival MSCs is what we are still seeing inside. And I would like to acknowledge Manipal Academy of Higher Education, so for our continuous support, my staff members, my lab members and my staff, and thank you very much.
Yeah, nice talk, Sandeep. Here.
Nice.
Yeah. So how do you create this periodontitis model in the animals? Yeah.
So, ma'am, brief anesthesia.
Yeah.
So, interdental bone is a place where we lose our bone. So, this is the structure where our tooth is, tightly packed inside our bone, in the sockets of the bone. So, when we place a bony tooth food material, so that is where it stucks here, and I open and I push it inside, deep inside, such that it won't come outside. So, post 2 to 3 weeks, we have seen the bone loss at the particular place where I have put.
Okay. And you have seen the potential of this GMSs in the animal model,
Yes,
right? So looking at the clinical perspective, so there's a potential to be used in patients. So do you think this stem cell transplantation using the GMSCs or the exosomes, do you get sufficient cells, or is there something you need to work more to use it in the patients? Is there any lacuna in this?
In patients, what we are doing is, for suppose we are taking bone grafts, like synthetic bone grafts. So it doesn't have immunomodulatory properties in that. It just fills the gap. So that also body has to accept. So if for suppose the hole is 100%, we try to fill it 100%, and acceptance changes from person to person. It may be 30%, 40%, still the problem will be there.
Okay. Okay, thank you.
The effects seem to be quite drastic.
Yes, ma'am.
So the GMSCs are autologous or allogenic?
So, ma'am, here we have used human cells for...
So it's basically a xenograft, right? So are your cells existing in the transplant site? Are they not rejected?
No, ma'am. They are not rejected. What I have seen is, so after injecting into the place, so I am injecting into the tissue specific defect. So we clearly saw that repository of the stem cells, maybe whatever we are transplanting, it is giving cues to the surrounding milieu, and it is helping the bone to regenerate there. And also the other property of stem cells is immunomodulatory effect. So the inflammation already persisted, that is periodontitis, that is reducing. So that is where we are seeing the regeneration. If the inflammation is persistent and we cannot see the regeneration, again the resorption will happen.
So have you sort of tracked your cells which you have
Cell tracking I have
transplanted
not
and
done.
checked what happens? Because it looks like very miraculous to have such kind of regeneration.
True,
So
true, ma'am. But with this, I couldn't track the cells. But, sir, please.
Thank you.
Sandeep, here.
Is it? Yes.
Oh, okay. Sandeep, carrying along with the question Dr. Manas asked, as per our knowledge, 25 million cells is the dosage, a single dosage for treatment of a human osteoarthritis patient, right? How did you calculate your numbers for the cells in the mice model?
Ma'am, here the space, interdental space is very minimal. So first I tried to inject with the Hamilton syringe how much volume it is taking inside, because more than that volume if I take also it will spill it out. So there I could find like 50 microliters is a maximum we can push it at one side, one time. So in that 50 microliter, I could constrict my cells around 2 million cells.
2 million cells. So considering you have a mouse model,
Yes,
the body weight would be somewhere around 200 grams,
yes,
right?
ma'am.
Roughly,
Yes,
roughly.
yes.
So 2 million
Rat
model. So 2 million per grams of weight is what you have calculated, and that has given you results, the ones
Yes,
that you
yes,
were talking
yes,
about.
yes.
Without rejection.
Without rejection, and also I have done only one time injection.
Okay.
So no multiple time injections first time. So first I wanted to see whether single injection is helping out, whether regeneration, or first of all, I'm not thinking about regeneration, whether my inflammation progression will stop or not, because I have created model both the sides.
Okay.
So one side it is progressing, other side it is started regressing.
Okay.
During all this process, you should have a controlled bone formation, isn't it? Overgrowth is also, because the bone size should be actually matching. There should not be overgrowth of the bone.
Sir, as such overgrowth.
If I see your crest bone in the treated one, if you go to the figure. Yeah. Next one. Next. Forward.
2 to 3 months old.
Yeah, here, is it that your ABC crest bone has overgrown in the stem cell injected one? Is it that portion?
Sir, overgrowth and all we have seen. Sir, if overgrowth means the space has to fill and it has to come out. So it is constrained still inside the gum only. So gum to bone minimum, like in humans, it will be 2 to 3 mm, which is ideal. So from the crest of the bone to the superficial gum. So that is called the pocket depth. It is ideal for everyone. So here we didn't see any overgrowth.
Okay.
Do you plan to use any marker to say whether the GMSCs that you injected is actually one which is regenerated, or any marker, if not, like let's say for example GFP transfected GMSCs can be injected, or a specific marker for your GMSCs to check whether they have, is that the one which has homed? Or is
Sir, homing thing I have not seen, sir. I have not tagged or anything for that, and to see whether only GMSCs.
That would help actually to prove
true,
that
sir.
it's actually GMSCs that has
True, sir.
induced it.
So for that, because for that sake only one side I have taken disease model, because if it is control and one side is control and one side only disease means that time there could be any difference, because one side disease is continuously progressing. Because let it be GMSC or any other thing, we try to put something external to safeguard that area. So that is only GMSC what I have injected. Yeah, sir, sure.
Sandeep,
Yes, ma'am.
what about inherent mesenchymal stem cells? The gingiva is quite active in proliferation.
Yes, ma'am.
Then is it that they are regressed in this condition? And is there a way that we can, inject some factors just to activate them instead of injecting xenotransplanting cells?
That's why we have narrowed down to exosomes. We have pulled first basic. We have seen whether cells have the potential, whether whatever the chosen cells have the potential. That clearly we have seen. Post that, as ICMR guidelines are there to bring this potentially to in a translation state, we thought of going for conditioned media therapy or exosomes. That we tried. So there also we have seen beautiful results. Okay. So because this can pave way for translational research. Yes, ma'am.
I think one last question. Can I look at the methodology slide? Yeah, the next I think, where the mouse model. Yeah. Okay. Okay. Looking at the thing, it's a 2 to 3 year old mouse. I have a couple of questions.
This is not 2 to 3 year old.
Okay.
This is
3 weeks.
Months.
Okay. Oh yeah. Okay. Okay. Yeah. Okay, coming back to that.
150 to 200 milligrams weight.
Okay. So basically, if I'm not wrong, understanding how you created the model, you inserted some food particle in between, so that after 2 to 3 weeks it develops. Have you ever tried removing that and see whether it goes back to normal?
It won't go.
Like, no, I'm just asking, will it, no?
So because with the saliva it absorbs.
Okay.
It won't be there.
Okay.
It dissolves, right? It's a food material, feed material. So till
So
it
how
stays-
long does it last in that socket or the place where you actually insert that? Since
Only
it's
one time.
Only one time. And within 3 weeks you see the...
Yes.
Okay, great, thank you. And one last thing, do you think the age of the mouse can actually impact how it's recovering? So right now it's at 2 to 3 months old.
That's why we have not taken younger also, we are not taken old also, we are taken adult rat. So ideally, because this disease periodontitis, ideally it won't be in younger ages. It starts at the adult age due to lack of maintenance. So if you try to interrupt at the early age, like in early adult age, it won't progress to older age.
Okay.
It's a progressive disease, right? By the time if I take old means it would have, because reconstruction and all will be very tough.
Okay. Okay, thank you.
Sandeep here.
Yes.
So we know periodontitis occurs only when there is continuous insult for a longer time. So that the model that you have created, there is only one time that you are putting the food material and there is only bacteria formation at that time. So how do you justify? You need to have continuous supply.
No. See, what happened is handling that, giving brief anesthesia. If we do multiple doses, we may not expect that rat to survive. First thing. Second thing is when we are placing it, I am not opening complete gum. Only what particular place, how much that is able to take. So we tightly pack in that, such that it won't come out.
So you are saying that for over a longer period or over a 3 week time.
3 weeks.
So the feed material is still there inside the socket that you have placed it.
Because I have when I saw all the animals, it is not there. It's already dissolved. It went off.
So that's what. So continuous presence of a food material, only then the bacteria will get it.
No, I'll tell you one thing. So though the food absorbed or it went off, the inflammation created or the infection created by that is there still in the gum, right? So that is helping in progressing the disease. It is not that the feed material has to be there or the thread has to be there in that place.
And when you're putting in the GMSEs,
Yeah.
so how do you ensure that the GMSEs is there in that socket?
Yeah.
Immediately after you put,
Yeah.
when the animal comes out of the anesthesia
Yeah.
and when it's eating and feeding, so there's a chances that the cells just get washed off, right?
See, while injecting into the place, I make sure that I know like where and all we have created. Sites is only one place, every animal, same place we have injected. I told the volume what we take is very little because that is the maximum it is taking, otherwise it is coming out, as you told, spilling out. So the injection, after injection, we wait for few minutes such that reversal of whatever cells or like whatever the liquid I have put inside, it won't come out. So that we make sure I am not excess anywhere.
So there are chances that the cells are moving across that area.
Once or twice it can happen.
No, there are chances that it's not localized to the defect area and it is moving around inside the gums or inside the bone that is already there. So localized that the cells are there for over a period of time has not been checked.
See, actually I'll tell you, for humans, for suppose, same this progression what we showed 2 to 3 weeks, so that will be years, like 5 to 10 years. So considering age of the rat, all those things, this 2 to 3 weeks itself we will consider in our human 5 to 10 years disease progression. Understood, right? So if you correlate like that, then that is ideal, because that is what I have seen different. After 2 to 3 weeks, for suppose if you wait for 4 to 6 weeks, through and through damage is coming. That also I have seen. And while injecting also, when we are trying to inject in that place, it is staying there. Staying means I have not tracked as such that one, but I understood with this multiple series of animals checking. So I felt like that has worked out.
Okay, thank you.
Yeah. Okay. Thank you. Thank you.
Thank you so much for that interactive session with all the speakers. And with that, we will conclude the flash talks. Now I would like to call upon Dr. Shweta to continue with the program.
So that actually brings us to the close of the program for all the talks. As we conclude the celebration of the National Science Day, I feel very privilege to deliver the vote of thanks. But first and foremost, I would like to extend our heartfelt gratitude to our esteemed guest, Professor Rangarajan, for taking out time from his busy schedule to come. And we thoroughly enjoyed your talk. You would have seen with the questions that were even asked from the students. I'm sure the students have been very much influenced by your words, so thank you again. I also thank our faculty, Dr. Vinay, for delivering such a great talk. And all our PhD scholars. This was the first experiment, I think, but it was quite a successful one, so I hope this tradition will continue. And the other enthusiastic volunteers that came forward, I'm sure you will be getting opportunities in the future Science Day. So thank you all again to the PhD students for making this Flash Talk a success. I would also like to recognize the dedication of our Dean, Professor Jyoti. Her leadership, her vision in organizing this event has been really key for me to do this activity for the first time. And I would actually like to invite her to give out some appreciation certificates for the newsletter. So we had a lot of participants who helped in making, you only saw the front page of the newsletter, but that's going to go live. You're going to be able to read the entire newsletter, and that has been put together by a team of students. I would like Professor Jyoti to give the appreciation to those students who actually helped the editors, Dr. Uttara and Anujit, to put that together. So first I'd like to call up Dr. Nasira. Also Jeevan and Abhinaya, as well as Trisha. Oh, and of course, last but not the least, Inchara. Thank you, Jeevan. I think he didn't take a pic, yeah. Abhinaya. Inchara. And Trisha. Thank you, Professor Jyoti, for coming up. I would also now take this opportunity for a special thanks to our office staff, especially Mr. Harish. You all know that he's always tirelessly working behind the scenes, without which no event can be executed smoothly. I want to thank the student volunteers, especially Janvi and Iona. I couldn't have done any of this today without their enthusiasm and support. And finally, all of you, the participants, for making this event super exciting and engaging. I'm really glad that you came forward with your questions. The last talk shows, you know, that you were interested till the last moment. So really thank you for that. And before we break for lunch, I'd invite you all to the stage for a group photo so we can, you know, remember this event for the future. Please, all of you come on stage.